All right, good morning everyone. And so before we leave, I'm seeing some jumbled rocks over here, and so maybe if we can just see if some of these angular rocks, depending on the size, actually I know you like to pick them up with the portmanip. Maybe that one that's sheared, is that too big?
Yeah, maybe we should, where is John here?
Oh, good.
I mean, how much of this is like some kind of rock fall with stuff coming from somewhere else?
Oh, it's all talus from the...
Is it, you know, if you zoom back out again for a second,
Yeah.
so you can look over to the left-hand side.
Actually, that platy one to the right.
Yeah, see all of that stuff in there.
Is that
It
good
looks
or
like
bad?
it all came from somewhere else.
Okay, so that,
What do you think?
But this would be a good place to get a rock.
is that, what do you think of that one,
That's
John?
a
So,
piece of a pillow.
okay, so we can pick
What do you think of
just
that?
about
Good
anything.
size, so
Is that good or bad?
it'd be okay. But maybe this. What about this?
You point at
platy
the one
over
that you
here?
want. Okay.
Oh, that flat one right there. Yeah.
I don't know.
Let me just
That
look
might
at
be
what's
hard to
below
get.
it there.
Is this?
Yeah.
Yeah, that one would be alright too.
Yeah.
I think that's kind of big.
Sorry.
That looks kind of big
Yep.
to get.
Yep.
This might be easier.
That thin
Grab
one?
it.
Okay.
And there's a bunch more up over in there.
Yeah.
Like there.
Do you want to wait? I mean, I just saw this
No,
jumble.
let's just grab
Okay.
one. Yeah.
I figured we ought to
Always get another one later.
get them while they're uncemented.
Yeah.
So before we do, I just want to look at this little creature here.
Oh yeah. That creature is a mysid.
No, not that creature. Oh. This
that
creature.
creature.
Yeah.
That creature which is a sponge.
No. It's
Oh,
not
that
a sponge.
creature. Oh, that's creature.
Hey, you
What's
got
up
some
with that
stuff
one?
on that one, Rob.
Oh, and there's a Nemurti in there too.
That's what I'm looking at.
Oh, okay.
Okay. Looking at that pink creature down there. Okay. So, you saw the rock that we're interested in there, Cody? This thin one?
Yes, sir.
All right. All right, so while Pyle is about to sample a rock here, we'll introduce everybody who's tuning in to see our video. There's a live broadcast from the Schmidt Ocean Institute's ROV Falcor. We're using the, RV Falcor. We're using the ROV SuBastian to explore the deep sea. We're on the Yomie Seamount chain, which is a string of volcanoes that leads from the far North Pacific and extends south. Today we're diving on Yomie Seamount. As far as we know, this is the first ever ROV dive on the seamount. We're at 42 degrees, 25.9 minutes north. And at this moment, we're at 1,520 meters depth. That's about 1 mile deep.
Perhaps I chose.
That looks like kind of a crumbly rock.
Well, I guess we won't take that one.
Yeah,
though.
let's skip that one.
Any others here?
Yeah.
It looks a little bit harder. See how quick right next to it?
That's interesting.
Want to zoom in right here on it?
Well, I think that's 12 rock.
Is it?
Yeah.
I'm trying to
Is that full of gas holes?
Can't really tell.
Got gas holes in it? Like right there.
It looks like
There,
consolidated
there,
sediment.
there.
between- Looks like it's just. It's like a sedimentary rock coated in manganese. Fake rock.
So you're not interested in?
No.
Yeah, we're not. We don't want it. Yeah. It broke up too easily. It's not real rock.
Could try
There's
this.
this toe right here.
I pointed it out before. You didn't like that one.
There's a
Yeah.
bivalve.
Well, you can try it.
You tell us which
Yeah.
one. Which one do you want?
Okay.
Big it is.
Will it fit? That's what I was worried about. Some
Sure.
of those fit the claw.
Yeah.
Okay. Camera zoom in, James. Alright. Actually, I'm glad you said that. I'm gonna go. We wanted to run a test for this before.
Yeah.
So let me run down to
Well, what's that?
Thank you, River.
That's great.
Boy,
Looks like it's got a couple of brachiopods on it, Wes.
kind of trashy rocks here. Find one with a coating on it and hold it together. Huh. Oh, weird.
Not sure what that is.
All right.
Yeah. That one.
Bypass that.
That one doesn't. Abort that.
What about this?
Like that
Yeah.
one, yeah.
Try that one.
That wasn't a rock at all. That was just a like coated sand.
From the last one, yeah.
Yeah, those two are just semi-consolidated
Yeah.
sediment.
That one that's acting more like a
That
rock.
one looks like a real rock.
Oh, there, now you got it at an angle.
So, you have somebody from Louisiana that says hello. The Facebook name is Betty B. I'm doing whipped cream.
Well, there's a lot of snot coming off of that. It looks like a dead sponge, but there's a. Yeah, that looks good. There we go.
That right there, Tortel?
Okay, rock one.
Rock box 1 or 2 as is most convenient. YOM 101-1.
101-1. Yom.
Yeah.
A Yomie rock.
What are you looking for? John, do you want the special discount? What are you tracing? Just raw.
Angular.
It's a good size.
Yeah.
I'm gonna give a quick zoom, Scott.
Okay. Maybe some little branching foraminifera on top, but I'm not seeing too much else.
Yeah, not too much.
Yeah, that's good, Cody.
Rocks
Good,
on the
thanks.
roof too
Yeah,
much.
it's a little bit of discoloration on the top. It's kind of
Yeah.
red. I think that's bigger than any of the other rocks we've gotten so far.
Yeah, it's good size.
All right, so we're sampling this rock. We'll do some geochemistry, perhaps some aging on it. We're introducing the overall cruise. We're on this mission to explore the bottom communities of the seamounts of the Emperor Seamount chain. We're looking to see where communities change as we are moving from the north, the Aleutian Seamounts, down to the Central Pacific. And if you are interested in more details about that, you can go to website schmidtocean.org, and get some more details on the cruise, see who's on board, the scientists, and what our objectives are. My name is Scott France. I'm a biologist from the University of Louisiana at Lafayette, and I'm sitting here with Les Watling, who's a biologist at the University of Hawaii. And we have several other scientists on board, ROV crew and ship crew, and we'll get to some more introductions later. And now we're going to do some science. All right.
Camera's yours.
All right, so we saw
So
when we were
sort of some
sitting,
stuff,
yeah, there's some colonies that are going to be to the upper left that I've got to look at. And that went in rock box 1, right?
Yes, sir.
You get that, Natalie?
Yeah, rock
Thanks.
box 1.
Thank you.
Okay, so I'm gonna come off the ground right here and
There's a curly eel
Yeah.
pouch for you. Yeah. It's interesting when they're young. I think they do that. Okay,
So right here, there's something,
take a look
Cody,
at that one. Yeah.
and then we'll
And
have a
there's
look
one
at
over
that
here on the
later.
left, Scott.
Yeah, these. Okay, yeah, actually, these might be even better. These
Yeah.
two right there. Thanks. I'm thinking those are the Walteria sponges, Les.
Yeah.
Oh, it's working.
Saw some pretty big bubblegum forward today.
Yeah, very big.
Yeah.
Up at the top of that feature that we were on. Yeah, these to me look like branching sponges. So,
Yeah, they do.
Cody, I don't seem to have control of the iris. Same sort of problem as yesterday. Thanks.
Okay, try to click the iris again,
Okay.
and you have your control of it now.
Yeah, the dial isn't doing anything for me. So I'm going to click it again, all right?
It's an auto.
Okay.
Back to it. Okay, now
So there's another sponge over to the side here.
Yeah, so I'm dialing isn't doing anything.
Let me see
Thanks.
if I can jump in line. I'll do is you want to land out right here and just
Sure, yep. I think I see a Galatheid crab on this sponge.
So these are the branched Walterias. Right?
Yep.
Yeah, that's right. Okay.
Yep. There's another, so I'm seeing two of these sort of munid Galatheids, one on the branched Walteria and then one on that euplectellid or euretid that's now off screen to the right.
Oh, is that what that was? One of that little vase one?
Yep.
Yeah. Okay.
It is, Scott.
Yeah.
Are you read it? Boy, this one's completely overgrown with things. So it's a dead skeleton. It's been there for a while. So what's this? The oh, is
What kind
that a
of
spider?
skeleton
Is
do
there
you
a
think?
spider crab on there?
Is that? You see how jerky that is?
Yeah, do that again.
That's. Huh.
So that's it.
All right, so now I've got the iris control. Thank you.
Okay.
Yeah.
All right.
Try to zoom out
Okay.
just a little bit. I just want to test it.
So
Yeah,
Okay,
it's,
so it's
so
done.
I'm holding it smooth, but it's jerking.
Now try that now. All right.
That's better.
That's that cross feed from one of these dials.
Okay.
So now you should have full control of your iris.
Okay.
Try
Thank you.
Let's just see.
Yep.
You do?
Yep, I do. Thank you. All right, what do we want to look at here, folks?
so close up on that Galatheid, I think.
Sarah, how is the performance? Is it seeming better today or slower or?
There's also a snail down there at the
Yeah,
base
we're
of
not
that
going to get much of a
sponge.
shot at it because of the way it's sitting. I
Yeah, I'll
guess.
look at the one that's on the other sponge.
Yeah.
Might be in a better
Yeah.
position.
But what's interesting is that when I do this,
Okay.
I might just move
Yeah.
you
Otherwise,
to-
that looks very similar to the
Yeah.
sponge that we saw last time.
Okay.
Um,
So, and
Cody,
we should
should I
get that
just
little
turn my
stalked.
camera?
Is that a baby Walteria growing under the rock there?
Yeah. Okay.
And then we should look at that.
As
Yeah.
long as your laptop's on the Wi-Fi, it's worth opening up and giving it a try. Because then if it, but actually closed,
yeah.
I restarted this machine.
Oh, wait a minute.
Shut up.
no, I think it
It's
is.
not machine, it's just a web.
I think it is, yeah.
Yeah, I think it is too. Yeah.
But it is a graphics display.
It's a very young one.
Yeah.
Yeah. And then down here.
So this is one of these
Oh yeah, that's what we
Lefroyella-like
collected yesterday.
sponges. Yep. It's
Yeah.
got all the anemones in
What
it.
are we calling that one?
Lefroyella?
What's that?
Lefroyella? Should be down. Oh, I haven't got it yet. Another
Okay,
call.
is it Lefroyella? L-E-P-H?
L-E-F.
Huh?
L-E-F.
Oh, L-E-F.
R-O-Y-E-L-L-A. And then I think, I'm not sure if I'm seeing several hydrocorals or bryozoans. I think these are bryozoans.
Oh yeah,
all of
those are
these,
nice.
there's lots of them.
Okay, Lefroyella with anemones, right?
Yep.
This actually makes it worse.
And then there's a view of
Yeah.
that munid crabs.
Is that a munida?
Yep. And then there was one more thing I noted. One of these sponges, kind of like what we were seeing on, I don't remember what dive.
Laptop real quick and then see, like, get yourself logged in and then swap over.
Where'd it go?
Well, see, so you're
Right here. So it's that one that grows.
Oh yeah. Oh, that one. Oh yeah. What should we call that? That's like a
Don't know.
snake sponge.
Yeah, but it was the same thing. They always seem to be just
Oh, yeah, look
growing
at
in a
that.
row like that.
Yeah. Linear.
Oh, look at that. It's actually growing over 2 rocks. How interesting.
Okay.
It's like a stoloniferous sponge.
Yeah. I'll use that in quotes as a name for it.
Okay. All right, that's good. I think we're fine here. We can go find something.
Yeah, there should be thumbnails, and that was one of the things I mentioned to her. I'll pass that on to Ari, because when we were doing testing before this cruise, I saw that on this machine, but I didn't see the same issue on my laptop. And you didn't see the same issue if you logged in.
Is that the purple thing?
I don't know. Oh, that's something coming out of the tube. We really want to have a look at this. So we saw a whole
that's
bunch
what
of
we
these
want.
tubes
Yeah.
yesterday,
That's the
but we
tube.
never saw what was sticking out of it.
Yeah, that's the tube maker. Look at that.
It looks like it may be a worm, but it might be a serianthid anemone, actually.
Yeah, I have a hunch it's a syrianthid.
Oh, fantastic.
Look at that. Yeah, that's the tube
Mystery
maker.
solved.
It is a syrianthid.
Yeah.
Oh wow. Yeah, we need a nice close-up view of that. But we'll never get
Alright.
it. We'll never collect that.
Do you want me to zoom out all the way on my camera?
Yeah.
Okay.
Rock-dwelling syrianthid.
Yeah. So I think we got a rock-dwelling serianthid in the Aleutians.
Oh yeah.
We were all surprised because normally serianthid, the common name for that is a tube anemone, and they build these tubes. You can see the tentacles sticking out there on the front side. They're brown, and then it disappears into this whitish structure. That's a tube that's made of mucus and some specialized cells that this anemone produces, and it fires and it binds the mucus, and it can also bind up
That's
sediments
spectacular.
and other particles from the environment. And normally they're buried in sediment, but here you
Look
can
at
see
that.
it's attached to rock. And I imagine our colleague Tina Molitsova will be very interested in seeing this.
Yes, she's watching.
Oh, she is? Oh, okay,
On
great.
YouTube,
She's
she
a
says
specialist.
two tiny brightcomb at your rock.
Yep, saw that as well.
Okay.
So
We
one of
don't-
the things that's a hallmark of the serianthids is
We
you
don't
see
have
these.
live chat. Is that right?
Yeah, no, we do.
Oh, okay.
Yeah, I'll
we
put it
we don't have it up here. We need
Yeah.
it up here.
Natalie is translating it. You see that large ring of tentacles. Serianthids have a second ring of tentacles inside that first one, but yeah, just
Oh,
one
okay,
more. I
there
don't
it
think we
is.
have to spend too much more time. I'm certain that that's what this is.
Oh, Tina's online. Excellent. Yeah, so Tina will enjoy that. That
She might be able to
syrianthid.
tell us something about
Yeah,
this.
maybe you can. Yeah, maybe you can say something. Who that is. I don't think there's any hope of collecting it though, because
No,
the
not
tube
here.
goes into a crack in the rock,
Yeah, it would have to be on
and
a loose rock.
yeah. So we've seen
All right, let's move on.
many of these tubes, but this is the first time we've seen one. What is the size of this? We need the lasers on there. This is the first one we've seen with an animal sticking out the front end. Let's see, here come the laser dots. So this is probably
Maybe
2-3
centimeters
centimeters in
across.
diameter.
Yeah,
Yeah, so it's not very wide.
it's good, Cody. Thank you.
Yeah. Yeah, that's
that's a
spectacular. Well,
cup
I'm glad
coral
we
on the
finally
left.
saw that. So,
Can I have just a
that-
brief look at that wormy-like thing up here. Thank you.
We finally managed to see who is in the tube. That's just really exciting.
This
Oh,
might
there's
be
a
an
pink
aplacophora,
Nemurian
maybe.
again.
No, you think that's?
Oh, there's a brachiopod. That's a brachiopod up there on the left, in the center.
Right. Okay, so that's a ribbon worm, stoloniferous octocoral, like we saw yesterday. That's the ribbon-like white thing that you see on the bottom.
So we got a
And
cup
then there's
coral
a-
over there too.
Small stalk crinoid, cup coral. Okay, we can move up. Heavily encrusted bottom here. There's an Anthomastus mushroom coral on the right side, that red one. We've seen several of those. There's several more Anthomastus colonies tucked in there. Let's have a look at this white fan.
Okay.
Thank you.
Oh yeah, yeah, Anthomastus.
A couple more of the munid crabs, squat lobsters that are on the sponge back there. I'm going to check to see if this is a primnoid octocoral or a hydrocoral, this white fan. It's looking like a primnoid.
Okay, Tina is noting that the Cereanthid collected from a sponge off Hawaii was a bit smaller, or a bit similar, it says, but hard to tell without collecting. Yeah, and I'm sorry that well, I don't think we're going to be able to collect that any of those because of the way the tubes are arranged.
I think I see an aplicofferan maybe in the background there. We'll get to that in just a second. But yeah, so this is a primnoid, this white fan. Looks similar to what we collected yesterday. And I'm just going to zoom in the background there and all of that cobwebby stuff.
Yeah, oh, that's interesting. Yeah, look at that.
Huh. That is not.
That's more
an
like
aplicofferan.
a
It's a worm, I guess.
ceratulid or a terabellid, probably a ceratulid polychaete,
Yeah.
but really hard to
Yeah,
say.
it is. Yeah, so you're
Yeah.
looking, I think, at a specialized worm whose
Yeah.
head end has all those tentacles that extend, and they can sort of creep out over the bottom, pick up particles, and carry it back to the body. And you can see that the body is very highly coiled.
Just watching stuff.
Normally, I would expect to see that in some kind of a burrow, not out loose like that. So that's really interesting.
Yeah, normally that, if that's a ceratulid, normally it would be in the sediment
Yep.
and in an area with hydrogen sulfide, actually.
Okay, we can move on, Cody. So we've just started our dive. We got to the bottom roughly half an hour ago, and we should be on the bottom here diving and hopefully streaming video for the next 5 or 6 hours. Let's have a look at that pincushion white thing upper left. Thank you.
Yeah, that comes better. Yeah. This.
I see. It's another sort of Walteria sponge, I think. Yeah, it is. Okay. We can keep going. Got it. Yep.
But then when I started capturing
Another primnoid here.
Oh yeah, you can see it. So if you just close that window, do it. But if you want, I can get you a cable and we can put your laptop on that screen. But it would
So
still be
lots of
running
Walteria sponges. So far, that's the sort of dominant thing that I'm seeing.
So, but the more alternative, but I can get you
Let's maybe go towards top center. There's something white.
this screen to be able to see.
I'm pretty amazed at the jumble of rocks in
I
this
agree.
area. It's quite a rock pile.
We can actually.
So in the sort of larger context, we landed in a little bit of a flat area and we're heading up a slope. So it's possible that this jumble of rocks you're seeing has tumbled down from that slope.
But it's easier to.
So this is another one of, I think, the Lefroyella type sponges. It's got a lot of anemones and zoanthids in it. That's good. We can keep going. Cody.
Put that on my list.
There's another one here, the Lefroyella sponge on the right. Then a Walteria sponge is this branching sponge that you see in the center.
Yeah, the Walteria branching sponges are just there to fool us because we would normally think they would be a bamboo coral.
Is this a
Okay,
spider
is this
crab
this
here?
Macroorgonia or? Oh yeah, it is Macroorgonia.
Macroorgonia crab.
The crab with the weirdest name.
And maybe after we pull out, we can get the lasers on just for a size for that thing. Okay. There we go. So those two green dots that you see next to the crab are 10 centimeters. So I would say the legs themselves are at least 20 centimeters, maybe a bit more. So half a meter across maybe when those legs are extended.
Yeah, the
impressive.
body itself is about 10 centimeters long.
Yep.
Yeah, so that's a good sized crab.
Yep. All right then. Sponges, sponges.
Now here's a stoloniferous on the underside of
Yep.
that rock.
Yep, that's the second
I'll
one we've
take a
seen.
good look. Do we see one of those
Yeah,
already?
when we were stopped down below.
Oh,
there was
okay.
one.
I don't have. I can check that off.
Yeah. Oh, let's look on the left side. I'm seeing a whip-like structure. Another Walteria sponge, I think.
The comment about whether the seamount is still active in any way because
Yeah, volcanism. Yes. Let me get a close-up on this first. So this is a nice-looking Walteria sponge. We've seen several of these. So these are glass sponges, Hexactinellida, and we can recognize them fairly easily with those short sort of spines that are sticking off the side. All right, that's good. Yep, Les. So these, as far as I know, these are not active volcanoes. They are something like 80 to 90 million years old and long dormant, as far as we know. So no chance for eruption today.
And no vents to see.
No vents to see.
Yeah, they're stone cold, I think would be the right description. Especially considering that the water is 2.4 degrees.
Oh yeah, yeah, it's cold down here.
Now here's where you can see where the rubble's coming from. See how broken up all this rock is? So there must be earthquakes out here.
Once you get it fixed.
The yellow things you see are feather stars. They're crinoids, related to the sea stars, or type of echinoderm.
But we might want to point out that all of these old seamounts were likely above the surface of the ocean when they were young. And in fact, we know that the one we were at yesterday, Suiko, was in fact an island, and there was a lot of pollen found when the island was drilled into, and the pollen turned out to belong to 5 or 6 different groups of ferns. So the island was covered by ferns, including tree ferns at one time. So those brachiopods in the on the rock surface there under that primnoid.
Yep. I'm gonna get a look at this, which I think is another primnoid colony, and then a couple of these little smaller white ones down there. See if those are more recent recruits or something else.
Who's in the bottom?
I had 2 brands.
Amphipods on this colony.
Oh yeah, look at that.
I know.
Huh. Very nice.
Yeah, it's just.
So this is definitely an octocoral in the family Primnoidae, and you can see the polyps themselves look very rough. Those are scales. So this looks like the one we collected yesterday.
Yeah, it does. It looks very much like it. Yep.
And lots of amphipods.
Oh, look
living
at the
on
amphipods have spines on the back. So, yeah, I think I might know who this is. This is one of the pleustids, I think, that we collected in the Aleutians.
Right, that's what I was saying yesterday. I thought that the amphipods were Aleutian related.
Yeah, I think so. Yeah.
All right, let's get a look here. The brachiopods that you were referring to. And then you see these very small white colonies. I want to find out if these are hydrocorals. Yeah, they are. Okay, so we got some hydrocoral also. You notice the structure is kind of different from what we were just looking at.
Yeah. And then there were some small white stalks down in the bottom of this pit.
Okay. So several of those. I don't know if they stay small or they get larger and just.
Yeah, good question.
Okay. What did you
Are
see?
those just sponges?
Oh, those are dead sponge skeletons.
Yeah.
Okay, that's good, Cody. Thanks.
That's quite a window on this world. That's amazing.
I think that's a sponge with a bunch of crinoids on it. Mostly dead sponge. You can see the sponges that are alive are much whiter, more transparent. The ones that are dead have more, the tissue is turning brown, and then sediment sticks to them, so that it also takes on a green-brown appearance, and then lots of other organisms will land on top of it and grow. There's something Yeah, we can have a look at this. Okay. This looks like it's covered in hydrozoan colonies, that sort of feathery stuff that you see. No space goes to waste down here. There's also a cup coral in behind it. Maybe some other very filamentous hydroids. The yellow, I'm not sure what the yellow is. Zoanthids, maybe.
Is there barnacles there too?
There's lots of amphipods. I'm not sure about
Yes.
this. Is that? Oh, the
Like
yellow.
this.
But is the yellow barnacle?
There's a lot of things on this sponge skeleton.
Yeah.
So skeleton is pretty useful for lots of organisms long after it died.
Okay. Over here, Cody, just tucked in down here, I noticed something red. Yeah, right there. Looks like it's another Anthomastus. So this is the one that's a slightly different color, I think.
Yeah, that's right.
We saw the red ones when we landed. I'm not sure if this is the same or different.
Yeah.
A comment from Tina asking
No.
how long ago.
It's got. Looks like it has a taller stalk than the other ones that we've seen. But
Scott, do you have a comment from Tina?
yeah.
What is John's approximation? How long ago? Oh, so I'm not sure, Tina. We think the age of the seamounts is some 80 or 90 million years, as far as I know. John's not in the room right now, so I don't know.
Right.
Do
Yeah.
you want lunch?
Oh, is it already that time? Yeah.
It's already that
Okay.
time.
Yeah, I'll go, Brendan.
Okay.
Thank you.
Zoom and stop.
Okay, that's good.
They must know if you can zoom at the stalk.
Can we zoom at the stalk? Tina wants to know. I think we can. See whether there's
Oh, she's gonna want it focused
siphonozoids.
too.
Yeah.
Yeah.
Slowly zoom at the stalk.
There you go.
That's cool.
It doesn't look like. I'm not so sure there are siphonozoids in on the stalk below the level of the polyps.
I think that's probably what she's looking at.
Yeah.
We have a little scale one, right?
All right, Tina, that's your view.
Okay. Did she say? Whoops. There's also Facebook. Facebook.
Yeah,
comments.
no, this is not currently active. We can move on, Cody. This
Okay.
seamount is not active, and I think it looks new because you've got this manganese coating that's been scoured, and so it looks polished.
Yeah.
But it's really not. I'm going to lunch.
Oh, lunch. Okay. All right.
You want to pass my water bottle in? Yes. Thank you.
Yes, sir, you do.
So, saying that if you want it on the big screen,
So we go around this
that
speed.
said that she could.
That's how you do it.
Yeah.
It's on stuff.
Alright.
Yeah, so that may have been an Anthomastus.
No, we got onto an area pretty smooth by the looks of it. Yeah, it's a young Walteria. It's nice to see the young colonies here because a lot of the larger ones that we've seen are dead. So it does suggest that there's been some recruitment, which is nice.
That over there.
Another sponge skeleton.
Let's take a look at that one on the right, see,
Yeah.
just to make sure what that is.
You see where we're going, Cody, right? You saw
Yeah.
it.
Right here?
Yep.
Oh yeah, it's the same thing. There's this
Yeah,
whole
another
class,
sponge.
this whole sort of age class of the Walterias that look like they're pretty old and have been dead for quite a long time, and they're very heavily overgrown with other things. Primarily hydroids, I would say, but also brittle stars and crinoids usually. So, and that one may have brachiopods on it.
Up there. Does that look the same?
There are more brachiopods here. We haven't seen brachiopods very often on the dive so far. Yeah, that's a really thick base. I don't know who that belongs to.
All right, Cody, we can keep going.
Moving on up.
Thank you.
So Tina's saying in Anthomastus you usually do not see individual siphonozoids.
Yeah, I'm not sure. Well, next time we get see one of the dead ones, we'll look a little more closely and see if there. I thought there were barnacles on the previous one of the previous ones, but they were sort of around on the back side, so I couldn't really tell. There's a very young Anthomastus near the bottom of the screen. There's another one coming up the right side there, the red one.
You want a shot of it?
Yeah, be good to get a close-up look. Oh yeah, I think this is one of the ones that we've collected that has the short stalk. Oh yeah, open up there. There you go. Yeah, the stalk is so sufficiently short that you can't really get an image of it from the side. But we should zoom in and look at the there you go. That's nice, man. We can zoom in a bit. We can look at the surface underneath the polyps. See.
Sorry, wrong direction.
Yeah, okay, yeah. All right. Yeah, now you can see the little siphonozoid bumps on the top of the. Yeah.
Good.
All right, yeah, that's good.
All right,
There we
Cody,
go.
we'll keep
Yeah.
going.
Okay.
Anything else?
Now, there's a lot of other white spots all over the rocks here. I wonder if we should take a close look and see what those are. See up in this lower sort of center left area.
Yeah.
Coral on the right.
Over.
Yeah.
Oh, there's a stick coral. Yeah. To the pan right. It's just there. By
There we
the
are.
pool.
See that one right there? You
Oh
want
yeah,
something
there's
on
a
that?
stalk there. But see the underside, I think that might be a stolen Ephorus octocoral.
That's the
Down
one.
your
Oh
left.
yeah, so it just went off the picture here, right over in here.
Where they're going.
Yeah. Yeah, there's
Yeah, we got
that,
that.
Okay.
but that's not what I want to look
Oh.
at right now.
We thought you'd be excited about a possible
Yeah, I
stick
want to
coral.
look at all of this right in here. Then we'll go back and look at that.
It's right under the sand screen now. The sand.
Oh yeah, no. See if we can.
Chris is dead. I for your ship second engineer's dead.
Oh yeah, well, I have to go in a little bit closer to see what those are.
Get my focus right so that we can.
Oh, maybe they are small barnacles.
This is, I think,
It's
bamboo.
Max.
Tina thinks that stalk was a bamboo.
We're gonna get to it. That's max focus I got. Good enough? Good, Les?
Yeah, I can't tell.
I can't get any better.
Yeah. All right. Yeah, I can't really tell what they are. I'm not sure they're barnacles, or I don't know whether they're, or are they, like on one of the rocks we collected yesterday, there was a little white thing that that was standing up, just a little thing like that, and we thought maybe it was the base of a little hydrocoral that had settled. But there's an awful lot of them here.
Yeah, there's a lot of them around.
So, yeah. So now we'll go look at that white stalk there, that one, which might be a bamboo coral. Yeah, I'm wearing my lucky bamboo shirt today, so I make sure that's what we get. Yeah.
Can we get the lasers on, Cody, please? Thank you.
Lasers on.
Perfect.
Yeah, let's get going. Can we get a little closer in on this thing? It's not very big. It looks like it's about 10 centimeters tall, which is about what the, oh yeah, definitely. This is about what some of the other bamboos have been that we've seen, but that's, yep, that's what it is.
All right, there you are.
So, yeah, and Tina also pointed out there are chitons on the rocks here, and we actually picked up a rock yesterday that had a chiton on it. So for this guy, this is looking, I don't know, can you focus on the lower part, on the, get the lower part in focus? Let's see. Yeah, it's got the ophiuroid on it as well. Yeah, this is, we collected two of these, but they turned out to be in, looked like they're morphologically in different clades. This one, it's got the C clade look about it. So I don't know if we should collect this or not. Maybe we should. Oh, there goes an isopod across the rock right in front of it.
You want to try and set up to collect it?
Huh?
Do you want to collect it or not?
Yeah, I do.
All right. It's pretty small. But
It's pretty small. It's 10 centimeters.
Is that an icidid? Is that an icidid? Is it an icidid?
Yeah,
Yeah, it's weird.
the one
the
we
way-
already have.
Are you sure?
Yeah, with the really long needles.
All right.
But we're gonna try and get this one too.
Yeah, that looks weird.
We'll go for
The pink inside the polyp was oddly distributed.
Tina does
you can just take it from the bottom.
take shot of bottom.
Yeah, I think it's small.
Yeah.
I think
The
it's gonna
structure of the
go
polyp.
right into your claw right there. And then
This could go in a quiver. Probably the
your
thing
choice
to
on
do.
quiver.
Quiver. So why don't you pull quiver out? Quiver and
Careful of this stuff. So this is going to be station 102, right? Depth 1482.
Yeah, this will be 102.
Time is 22.
There's a rubber on there. Clear. Is there? Just clear? Yeah.
On for quiver number 8.
Okay.
Number 8.
Do you need a different label other than that right now?
Yeah, that's fine. Yeah.
So, Jessica, this is station 0102, sample number 1.
Yes.
Going into quiver 8.
Yeah, I'll just mention also while we're doing this that one of the rocks that we had yesterday did have a chitin on it, and so we have the chitin and we have it preserved, and it's not very long. It's literally probably 5 or 6 millimeters. So I don't know if that's true for all the chitins here, but that one was particularly small. But I think that what we can do also, though, is probably get a nice close-up image. I think we have one actually from yesterday, but today we'll get a nice close-up image also, and then see if we can send it off to see if we can find out what it is.
You rotate around 180.
Just try it out.
So these bamboos are a little bit shallower than the 2 that we got the other day.
I've got a good feeling about today. I've got a good feeling about today on bamboos.
Yeah. Oh, see, I wore my main shirt today.
Yeah, excellent.
Did a lot of bamboo collecting when I was under my Yomie guys, so
All right.
I thought that's what I would do.
That'll work.
I was just gonna ask if you do it in the office, so I wasn't sure.
Yeah, I guess.
So we're momentarily paused to do a shift change. So new pilots and co-pilots switching out.
Not too bad. This is the co-pilot console now? It's going to be $10.
Yeah, it seems good.
So, I guess we'll be thrusting back.
Yeah, when I try to grab it's being pushed down.
I think it's good. Right now, I gotta go thrust forward and a little bit down.
I'm gonna reset.
Yeah, in response to Tina's comment about the rarity of chitons, I think that's by and large been my experience on with all of these seamount dives, that chitons are quite rare. But we have seen chitons now on both of the seamounts that we've investigated so far on the here in the Emperors. But what's different about this seamount versus the one yesterday and the day before was that this one also has a lot of brachiopods, which I don't think we saw at all on Suico seamount. So I'm not sure what that difference is, but this is a pretty tiny coral, and it's kind of tucked down in the bottom here. So I don't know whether this is going to be easily obtainable, but we'll have a look.
Yeah, they're gonna get it.
Sneaking up on it. Skittish. Lunch?
Monday.
I just say it louder.
Okay, good spot.
I don't make them very big up here, do they? This much.
Do you know, so you write to Lawrence, the Microbial, he will tell you what this is. I can hear you a little bit, but not all of it. Got it!
There's not going to be much for genetics. Well, there might not be much for morphology.
There you go. Now you said it.
Yeah, so I'm seeing a couple of images very important about it next week.
No, I'm okay. I have like brownies that had.
One there.
Yeah, this is your diet. So it kind of comes up and follows along. The weight might want to decrease at the top.
What's that elevation here? Pretty good here.
Yep. That'll drop. Q12, YON 102-1.
So it goes from 1500 to 1340.
We have a comment on the tubes that you're seeing here. These were designed by the pilots coming out of Honolulu as a way to allow us to collect a number of samples that could be kept isolated from one another. One of the interests in the samples that we have is to look at the microbial community that's living associated with these corals and sponges, that is the bacteria that live on them. And so as much as possible, if we can keep those samples separate before they get onto the deck of the ship, that way we can ensure as much as possible that the microbes that we are looking at are indeed associated with the coral that was collected.
Dad? Yeah, there's a message appearing on the wire class. And feedback is
It's funny that you wrote bamboo with,
Yeah.
or excuse me, you wrote bamboo, I wrote
Yeah,
it with.
yeah.
So it's funny you wrote bamboo as opposed to bamboo coral. I got a manuscript review request
Uh-huh.
because you're an expert in this field. and it's on bamboo, the plant. Like, I am not an expert in that.
Tina's asking about the tubes that we're putting them in.
Yeah, I just described that.
I can't hear everything you're saying.
Okay.
So.
Yeah, I
Does it
saw
help
the
that?
comment.
Yeah.
and
Does it help that I tell you the comments, or you want me?
Yeah, no,
I didn't know. If
certainly
I
one of, yeah.
How you doing?
I'm doing well, how are you? Sorry.
Because if it's being too repetitive.
No,
if
that's
it's,
fine.
okay.
Because I don't,
You're not looking up.
when I'm actively talking, that's something that it does help.
Okay.
Thank you.
So she asked you another question too. How do you prepare them for microbial communities or community?
Becca Lensing is a graduate student at the University of Hawaii. She is the person on board who's responsible for preserving the samples for the microbial analysis, and she's doing a couple of different things. She's scraping mucus, literally scraping mucus off of the colonies when they come up, and I believe she may be trying to culture some of those. So she's got some media here that she puts stuff into, and I think we'll see if it grows. And then some of the material is frozen. There's also tissue samples frozen and put in ethanol. So there's a variety of different ways to examine it. Ultimately, they'll be doing DNA sequencing and looking for signatures of known bacterial groups, and you can analyze the samples in that way. Cool. I see we have somebody from South Carolina, Peterson Geochemistry Lab, tuning in to learn more about these deep sea communities on the Emperor Seamounts. One of the things that I always find fascinating is, for those of you who don't have a lot of experience with deep sea science, deep sea biology, and maybe are just reading a line or two in a magazine or a book, is the amount of work it takes to get any sample. And you're seeing that here. I mean, it's not just a matter of, oh, I'm just going to pluck that. It takes a lot of care. In addition to what you're seeing, the pilots manipulating the vehicle and the manipulator arm to collect that sample, then to stow it and to stow it securely, you also have to keep in mind that this vehicle that we're working from is at the end of probably close to 2 kilometers of wire that leads up to the ship. And here on the ship, this is a pretty good day. We're not moving very much. We are very fortunate in the weather. Not too much wind, not too much swell, but some days the ship can be bouncing around a lot. And then of course the ship crew have to deal with the surface current and the wind, so they have to keep the ship headed in a particular orientation and try to have it as stable as possible so that this ROV can sit here and not get pulled by the wire. So a terrific sample secured in our quiver, and we'll soon be ready to move on and see what else we can find here.
Two end members dancing around each other on a 2.5 meter or 2.5 kilometers of cable.
Yep. Not an easy dance.
To sample a 10 centimeter
Yeah.
bamboo
Yeah, good
coral
point.
stock.
Yeah. That's maybe 2 millimeters in diameter. So
All right, you ready to take off?
I am ready to take off.
Camera is yours.
Camera is mine. Thank you.
Auto, iris, and ISO off.
Thank you.
Control of the ISO.
Okay.
Let me know if you want to decrease the ISO.
I'm just going to play around here, see if it's actually responding to me. And I don't see it responding to me. We had this problem earlier.
Okay, just try turning your ISO or your iris map.
Yep. Oh, maybe not. Nope.
Here, all right, let's try this.
While you're going there, J-Rob, we're going to want to have a look at this. I think it's a black coral.
You guys all right now?
Okay. Yep. That's good. Thank you. So we're approaching this sort of orange-looking colony. This is a black coral. We sampled one of these yesterday on Yomie Seamount, and it's interesting that we have following us in YouTube chat box Tina Molitsova, Russian biologist who's something of an expert on this group, and so maybe she'll have something to say. We thought maybe this could have been the genus Dendrobathopathes. This one looks like it's in bad shape. It looks like it's alive at the edges and dead near the center. You'll see as we get a closer look here. The live tissue is that orangey color, and the skeleton itself is black, and you'll notice there's no live tissue in the center. And we saw that on one of the colonies yesterday as well. Looks like it had been damaged and being attacked from the center up and outwards. And I believe I may see an aplacophorine mollusk. We'll verify that a little bit more closely. No, that's a polychaete worm. All right. So there you can see the skeleton denuded of tissue on the right side. It's kind of black, and then that sort of brownish stuff is a combination of mucus and sediment, and there might be some other organisms growing in there. The pink thing you see is a polychaete worm, and then we have this orange tissue on the left, and that's the actual black coral. And yeah, it looks like there's a snail in behind this colony. And Tina, that's the best zoom I can get you right now. Let's stand by and see what she can say about this branching pattern. Excuse me. The black corals themselves are actually described based on the way the branches, how many times they branch, and then some ultra-fine detail of the spination of those branches. They're also called the thorny corals. Alright, but this is definitely the one we collected yesterday. So we're going to move on. Thank you. There's one of those eel pouts curled up in the water column there.
On your top right is another whip-like.
Yeah, let's check that out right here. Thank you. Probably similar to what we just collected. And one of the things that's interesting is if we see that they're all this small size, that tells us something if we're never seeing any big ones. And I think I heard Les saying on a couple of dives ago we were seeing these whips, and they were also quite small. So the question then becomes, is that because there's something that limits their growth? Is that a genetic thing? Is it an environmental thing, something about the amount of food that's available? Or is it saying something about how recently they colonized this area and they're relatively slow growers? So there's no question that that's what we're looking at here. Focus up a bit on this. You can see those very long spines. Long needles. All right. Is this? Are you? No. This.
Are you working on your focus?
I am trying. Yeah. So I've lost the focus control of. Oh, there we go. There we go. Thank you. All right. So yeah, that's the same thing we just collected, and the interesting thing is there's needles just restricted to the tips. One of those may have some food in its gut. The polyp that's second from the top on the right. Okay. And then there was
Same
another
size
snail.
as the one we collected too.
Yeah, yeah, really
So-
small. We're good to go, Gerard. Excuse me.
Then you don't have to, you just set up a watch.
Let's have a look at this thing right here.
On my way.
Copy that. I see a sun star in the upper right of the screen. Is this maybe an Umbellapathes black coral? Or I'm gonna just get a sneak preview. This may be the same black coral we just looked at, not Umbellapathes. I take that back. I can hear you though, that's good. Yeah, so this is the same one, but we're seeing it from the other side now. You can sort of see that it's bowed somewhat, so it's curved, kind of like a catcher's mitt, if you're American, where the bowl of the depression is on the left side. And are the currents moving from right to left or left to right? Left to right. So that makes sense, yeah. So the polyps are basically downstream. So the water sort of flows through the colony, probably creates some eddies that return the particles back towards it. And then we have a Lephraella sponge on the right, more brachiopods. And then what did I see when we were landing? Nothing, I guess. Okay, we can move on. Thank you.
What appears to be a sponge or a fan off to the left.
Oh, yeah, that looks like a fan. Let's check that out. Good eye. There's another chitin here on the right. I heard you talking about the chitins earlier. We saw lots of those yesterday. These mollusks related to the snails that scrape the bottom with their teeth.
There's some other ones out.
There's an Anthomastus, I think, on the wall there. A couple of them. Oh, that's a sponge, I see. Yep.
There's another black coral.
Yep, another black coral. The sponge looks like that Lephraella, so we can skip that, the white one. We'll just check to see if this is the same black coral. I think it is. Another Walteria sponge, which is that erect sponge on the right side. Sure enough, this is again this highly branched, kind of planar but slightly bowl-shaped black coral.
We have a little primnoid on the right.
Oh yeah, primnoid, and there's a crab underneath it. Let's check that one out and see if that's a different primnoid. Okay, thank you. It's a fish tucked down there in the rock.
So Tina, but the black coral, quite possibly something like Dendrobathypathes.
Oh great, yeah, so Dendrobathypathes is, Tina, what I suggested yesterday it might be, so I'm glad to hear you confirm that. And Tina just made an interesting observation. So she's a deep sea biologist, she has a lot of experience, and she just said she's never seen it alive, so she's not sure. And this is one of the great things about having a tool like this ROV SuBastian. It allows us to come down here into the deep sea and see these animals in their environment, see how they live, how they interact with the environment. You know, are they growing on the side of a rock, on the top of a rock, in a high current? What other organisms are associated with them? So this is extremely valuable stuff, not just getting the collection, but being able to orient it in its environment. There you get a nice view of that chitin on the right. And then the brown thing that you see is a skeleton of a dead sponge, and there's a couple of brittle stellar arms sticking out of there. And then the white thing on the right, on the left, that's a octocoral in the family Primnoidae. We've now seen that a couple of times, and we collected one of these yesterday. And then the pink thing on it is an anemone that is living on the coral. And this is something we see quite often. A lot of these organisms that grow attached to the bottom, we call them sessile organisms, become habitat for other organisms. It's much like the trees of a forest. All right, good stuff.
Tina would like some pictures of the outline of the black corals from yesterday.
Oh, okay, thank you. Yeah, we can move on, J-Rod. I'm just going to tilt the camera down so I can see how I'm oriented with respect to the porch. Oh, I'm way up. Whoa, there it is. Okay, well, it's pretty good.
Those see the little white down there?
Yep, all this little stuff that's scattered to the bottom.
More to the left. Aren't those like a really old thing that are related to like
Yep.
clams and scallops? Or like
They're brachiopods. They look like clams and scallops, but they're actually not related to them at all. They do have a bivalve shell, so much like a clam, there are two shells, but the shells are made they can be either protein or they can be calcium. But when they open up inside them, there's actually a filter feeding organ. It's kind of horseshoe shaped. It's got a lot of flagella on it, basically, and they beat and generate a current. So quite different inside than a clam. But exactly, they have a really rich fossil history. So very well known in the fossil record. Many more species, I believe, known from the fossil record than are known alive. So good call on that. Onwards.
onward and upwards.
All right, you're good, right?
Yeah, thank you. Enjoy your lunch.
So if I can try to get up to like 75, 35.
And Tina will be happy to send you some deck images of the collected black corals.
Coral in the middle of the screen.
There's so there's a primnoid, the white one, and then there's a couple of yellow feather stars. And that the hydrocoral, I think it's what just went off the bottom. Boy, so what are all these little dots? Let's just verify what all these little white dots are on the bottom, because they suddenly increased in frequency quite dramatically.
Just like right here.
Yeah, everywhere, all this very small white dots.
I'm just trying to get to a pretty close.
Okay.
That way you can, and then
Yeah, so the question, and you see this thing? I think I'm going to want to look at that. So that's another solid sea cucumber. We saw one of these yesterday, so that'll be an interesting observation. Okay. So the first thing I'm looking at are these very small white dots that are on the substratum. Trying to determine what they might be. Extremely abundant. Oh, so they're very small recruits. We actually collected one of these yesterday. I think they might be little hydrocorals. I don't think they're forams, but I could be corrected. But the good thing is yesterday we collected a rock. There was one of those on the rock, and we did preserve it, so we can look at that more closely. Now I'm going to have a look at this thing up here. This is an unusual animal. We saw this yesterday. This is a sea cucumber, so it's a type of echinoderm. So this is related to the sea stars and brittle stars. So you can see a brittle star up top of the screen. That's an echinoderm. This is another group of echinoderms. So this is a sea cucumber that has become adapted. It's basically flat on one side. Most of the sea cucumbers, if you're familiar with them, kind of look like a sausage, and they are on soft sediment. This one has flattened one side, and it kind of suction cups itself to the wall. And then what you see there are those long arms. Those are actually highly modified tube feet that are branched, and they have mucus, and they can pick particles out of the water and then eat them. And what you can see is the lowermost tube foot is curled into the mouth. It's kind of like sucking its finger right now. Disappears in a little hole. So that's a sea cucumber in the family Solidae. Oh, there it is. It's sticking it out now. It's pulling it out. Finger licking good. And now you'll see that'll sort of uncoil and it'll be branched just like those other ones around it. That was well timed. It's having lunch just like we were.
so will it more than likely take in another arm?
Sure. Yeah, I mean, if it's got particles on it. There you go. So that's a good example of what a suspension feeder's doing. It needs some kind of tool to get out into the water, capture particles from the water, and then transfer it to the mouth. So all these things that you see attached to the bottom are doing something similar to that. Fantastic.
There's another one just up and to the left
Okay.
that I've seen before, and they didn't know what it was.
Oh, look at this. There's a solitary hydroid. This is in the genus Candelabrum. Bizarre. These are so bizarre. So this is a kind of cnidarium. I gotta try to get the focus a little better on that. And this thing is tucked down into this little hole. And think of kind of like a little coral polyp or an anemone. It's attached at one end, it's soft and it's long. And instead of having all the tentacles around its mouth, all those little balls that you see are short tentacles, and the ends of the balls are just packed with stinging cells. And if this thing were up on the rock and there was a current going by, it would look like one of those crazy things you see outside the car dealership.
Yeah, windsock kind of
Yeah,
thing.
like a windsock. That's a good description of it.
They didn't know what it was.
White balls.
Yeah.
Because it kind of appears like an anchored siphonophore.
Yeah, yeah, that's a really good description. They're very closely related. I'm not sure, Natalie, what the balls are. I'm not sure if that's something that it's captured. Nice.
And I think there was one more thing I saw from the zoom out.
Oh. There's another, looks like an anemone tucked into a little hole on the top right. Okay. Sorry, went a little too fast there. There's a brown sponge. Yep, with the brittle star on it. Yep, and then there's a cup coral in the top center.
And we got a coral and sponge.
I think that's a sponge on the left, Natalie, if that's what you're referring to.
Yeah, but it looked like there was a coral on top of it.
I'll check that out. Yeah, so that's a cup coral, sort of top center. Two cup corals. There's one farther up there. Yeah, I don't see the coral, Natalie. There's an anthomastus further up.
If you go down on top of the sponge.
The sponge in the upper left?
No, the one
Oh.
that's below your science tab. Down here on the right. Right at the bottom.
You mean the this
thing.
primnoid, the white?
Yeah.
Oh yeah. That's, I saw that when it came in. That's the primnoid that we looked at.
It is actually on the sponge.
Oh, you're right. Yeah. So I guess that's not a primnoid. That must be a stoloniferous one growing on a sponge. Yep. Way to be insistent. That's good. Yeah, so this is another kind of octocoral we've seen
Another
earlier.
bamboo.
This might be worth collecting because we see these grow on the rocks like ribbons, and you can't get them because they're on the rock. But this one is growing on a sponge skeleton.
Is that one colony or multiple?
That is one colony. It kind of grows as a ribbon and puts up individual
Like the one that saw on the
polyps.
rock yesterday?
Yep, that's exactly right.
Oh, okay.
Those are caprellid shrimp. It's an amphipod. So on the upper right, you see it's also called a skeleton shrimp, and it uses its back legs to hook on to the branches. And then you see it looks like a little praying mantis. It's got a couple of claws, and you can see its head and its antenna sticking out. And then there are more regular amphipods below it to the lower right. And then I'm not sure what the yellow fuzz that's overgrowing this colony is, but the stuff that the caprellid amphipod is on is called a hydroid, hydrozoan. Oh, and there's maybe a little aplacophorine worm, so that's a kind of mollusk. It's related to a snail. Where I'm looking is the top center of the screen, something that looks like a corkscrew wrapped around a branch. That's a kind of mollusk related to a snail, looks like a worm. And it's a specialist predator of cnidarians. There's all kinds of good stuff on here, so this is definitely a collectible.
Tina says take
Yeah.
them all.
Yeah, I'm sure Tina says
There's
that.
some long, thin, unbranched tentacles were sort of waving around in
Yeah,
amongst that
so I'm
group
not sure
there
if that
too,
was
and I
an
wasn't
anemone.
sure what that was
Yeah.
from.
Maybe if we can like take it there so we get
Yeah.
that piece.
In a quiver?
Sure, yeah, because that would nicely contain all that little stuff. Thank you. All right, so we're going to sample this. There's a lot of stuff on there to be seen. I'm turning over the camera to you, Gerald.
Make them all. I agree.
Our depth is 1,472 meters, and the water temperature is 2.4 degrees Celsius. That's about 36 degrees Fahrenheit or so. That's pretty cold.
Yeah.
Line. Just coax it out. So that's how you try to get your.
It's cold. You wouldn't want to be swimming out here.
I mean, you can try. I just think if you think it all underneath that. Close it out. Whatever you
So Natalie, the primary collection name is Stoloniferous Octocoral. I'm
Okay.
going to say it's on sponge with all this accessory stuff.
With P or with F?
F. This is the Stoloniferous. So I had caprellids, regular amphipods, aplecophoran, hydroid,
Hardwood, yeah.
sponge.
A lot of stuff.
Yeah.
Yeah. Extract the lid.
So we're going to put that one in a quiver. We might
Yeah.
want to try to get a filter or something in case anything falls off when we dump that out.
Yeah.
That aplecophoran was pretty small,
Yeah.
and they dislodge.
That aplecophoran is going to be very small.
Yeah.
Yeah. Tina says, collect the zoo. And a good way to express it is
Oh, the zoo.
Definitely
I
a
see.
zoo.
Yeah. Menagerie.
the menagerie. Yes, that would be. Yeah.
Yeah, before that you said collect them all.
You want like just that one branch, right?
Yes, exactly. It's the white stuff that I'm really going for. I guess I should note the hydroids.
How old is this rock? Yeah, I would say this rock is somewhere between 80 and 90 million years old.
Yeah, we've been having quite a few questions and comments about the rock, and earlier folks were wondering, you know, how recent this looks like a new volcanic eruption. And I think you're being kind of fooled. You're not actually seeing the basalt that was formed when this volcano erupted. Instead, it's been in place for tens of millions of years, and it's been coated in this manganese crust, and that's what you're seeing, and that's what looks so shiny and new. So just as a reminder, you may be following us on the video on YouTube. You can also follow on Facebook. You can learn more about this expedition at the website schmidtocean.org. We're on the RV Falcor, which is a vessel from the Schmidt Ocean Foundation Institute, the Schmidt Ocean Institute. And we're out here on the Emperor Seamounts on an expedition. We're at sea for about 32 days. And if all goes well, technically and weather-wise, we'll be diving over the next 8 to 10 days. Yep, that looks good. That'll drop.
Another 10 days, yep.
We're going to be making our way south along this Emperor Seamount chain. Today we're at Yomie Seamount. Very nicely done, Zach. That's in Quiver 8, Q8.
1031?
1031, yes.
Yeah.
Stand by while we fill out our sampling paperwork.
Now, just to the left,
Yep.
there is,
This thing?
yeah, that looks
Yeah.
like a verrucid barnacle.
Huh.
And maybe that's, so we should get a close-up of that.
Sure.
We were wondering earlier what all the little white dots were.
Yeah, so I zoomed in on that. That's
Yeah.
the thing that I took off the rock yesterday.
That's what I was
they are.
thinking, yeah. But there seems to be an awful lot of them.
Oh, there's a huge number here.
Yeah.
Huge number. And so I
So
thought
we were wondering whether they were also small barnacles, but it doesn't
No,
seem like
they're not.
like they are. But that particular thing might in fact be a barnacle. We have verrucid. The verrucids are one of the older groups of barnacle, of sessile acorn style barnacles. and they're quite common in deep sea environments, but we don't see them in shallow water at all, very often at all.
I tried having a look at the one that we pried off the rock yesterday, and I couldn't determine if it was a calcareous foram or a hydrocoral recruit. But seeing the abundance of these, and they're all the same size, I'm inclined to believe it's a foram.
Yeah, that's right. There's the numbers are quite large for a recruitment event.
I agree. And the fact that they're all the same size suggests that it's not getting any bigger.
Yeah.
Good afternoon, Gerard. As you can see there, we've had a little bit of a shift in the forces out here. We're still looking at supports. I brought the head around a fraction to start, but I may need to get around a little bit more. I just wanted to let you know that we're just finding new balance.
Understood. Yeah, just keep us up to date. I've been watching you. You're not pulling this in also.
Right, thank you.
Did you notice the solid sea cucumber when you came in?
I saw that.
Yeah.
Solid number 2.
Yeah.
Yeah.
This one looked a bit different from what we saw yesterday. I think.
Yeah, but I think it's probably the same one.
Okay.
Yeah. But you're right, it did look a bit different, but I don't think there's much species diversity in the solids.
Known.
We hope.
Known.
Known, yeah. That's very true. And they're very difficult to collect, so
Yes.
yeah.
We might be able to suction one in the right position.
Yeah, if it's out far enough, I think we probably can, because they do pop off relatively easily.
Yeah, we
I know collected one in shallow water with an old shipwreck grab, which is a spring-loaded grab.
Yeah.
And I didn't know we had lowered the grab into a rocky area, and it obviously bounced off the rock. But in doing so, it popped the solid off, because the only thing in the grab was the solid.
All right. There's a couple of things I'd like to look at before we leave. I'm going to zoom in on this, and then I'm going to zoom in on that.
Yeah.
All right. So first,
a little bit closer by bringing that ram in.
okay. We may not need to, but I will certainly ask you if we can.
There's a
identify it.
brachipod from the side. Yeah, that's what it is.
Yeah. Okay. So that's another one of the brachiopods. We're kind of looking down at the two valves on the left and right that are closed together. So that line you see down the center is where the valves come together, and they have a short stalk that they live on, a muscular stalk called a pedicle. And I think there's another one of those worms down in there that we looked at earlier. Yep, there's an open one down on the lower lower right. And then all those tentacles you see in there, I think earlier we were having a look. They come from
there it is,
feeding worm. And I just want to check, I wondered if I saw a cladorized sponge over here when you were collecting. Yeah, right there. Oops, sorry. Whoops, sorry, wrong button, my bad. I meant to be focusing.
Is that a little carnivorous sponge?
That's what I think.
Yeah, so that's what I'm guessing, yeah.
Sorry. The buttons have been reversed here on me. So
Yeah, it's very small, but
change my brain into knowing when I'm in focus.
Keep me on your toes.
Yeah. All right. So that little stick that you see in the lower right corner, that's kind of white, that is a sponge. It's a carnivorous sponge. So it has little Velcro-like hooks on it, so that it can capture things out of the water column, and then, believe it or not, its cells re-aggregate to cover up whatever it catches, and they digest it. Sponges don't have a digestive system. They don't have a stomach or anything, so that's an unusual mode of feeding. Yep.
I want to look at that.
All right.
I'm wondering whether there's barnacles on that brittle star.
I think it's in front of the brittle star.
Yeah. What the?
I'm not sure those are barnacles, actually. Those look like little hydrocorals.
Oh yeah, they're little hydrocorals. That's
Yep.
what they are. Yep, that's right.
So there's
They're in
three
front of
things that you see.
the brittle star. That's what.
Okay.
All
We're
right.
good to move.
Good. of the beauties of having an ROV with a really camera lens is that you can see how close in we can look at things in size and get a pretty detailed look at them.
Stand by, Jared.
So there's the immersion again.
No,
And
yeah, I guess it is. Oh, yeah.
Yeah,
See the coralled
yeah.
interior.
And then the brachiopods opening, living in it, actually sticking.
And very often when these brachiopods, the two valves are different sides.
Yeah,
And that's a...
somebody's.
Okay, I'll take the camera.
Tina has a comment, but go.
Yeah, forams as well. Tina, we think that's what all these little white dots are. I think they're calcareous, single-celled organisms related to amoeba. And some of them are able to produce a shell made of calcium carbonate, and in this particular case, attach that to the rock. I think that's what's going on. There is a comment, a question about whether we can move rocks to see if there's worms underneath it. There's certainly probably stuff living in the sediment, and occasionally we will move a rock because we're interested in sampling one, but we don't have the time to be poking around under all the, well, not all the rocks, but there's plenty for us to see on them.
Take off?
Yes, sir. So we will resume our course, moving up slope on this ridge. Copy that, Natalie. Looks like another priminoid colony here.
You haven't collected it
Yep.
from some of this died yet.
We haven't. I think we collected this one yesterday, but I agree, we haven't got one today.
There's a chitin. We want to get a close-up image of the chitin.
Okay.
Um,
Is it different from what we
They
found,
suggested we could send the chitin image to somebody and get it identified.
just off the bottom of the screen here. Whoops. So right there, that thing, and I'll probably launch. Oh, that's just a sponge. That's okay.
Oh yeah, she just sent the message with the name of the person.
Yeah, and I think I got a pretty good view of this chitin earlier when we stopped to do the, I forget what it was we were zoomed in on, but I was describing a bunch of things.
Yeah.
But we'll do one more, and uh-oh, there's our first urchin. Yesterday we saw literally tens of thousands of these urchins.
Yeah, that's right. Today we see one.
And can I have the lasers on briefly?
They are on.
Oh, they are on. Okay, so I got to come out a little bit. There they go. So that chitin is maybe 4 centimeters in length.
4 centimeters, so that's a lot bigger than the one that we collected.
Yeah, the one we collected yesterday was maybe a centimeter.
Yeah.
From this distance, we'll have to move.
Okay.
Stand by.
You can see some striations on the shells.
Yep, absolutely.
And well, that's a good, nice focus and light, because now you see the pebbling in the mantle.
Yep.
And those will be helpful characters, I think, for somebody to identify this thing. And it's got lots of stuff growing on it, so it's probably old.
Yep. And then I am going to, while we're sitting
Okay,
here,
I
I
gotta
will
get
look
the-
at this priminoid
urchin
colony
log in
on the right.
Less?
Yeah, so this is a really nice view of your amphipods there, Les.
You can see the It looks like this. Yeah, now you can see the spine back end of the thorax.
Yeah. Okay, good to go.
And then there was a black coral.
Yep, this one. It might be the smaller version.
Less? Eyes like the Chandra's?
No, I don't think so. Yeah, I don't, but I don't really know. I don't think so, though.
Yeah, so I think this is, you know, what we were calling yesterday the wall of black coral. This is a few times now. So today, this image that we're looking at right now is kind of the back side. The other images we've been seeing, the front side where the polyps are arranged. Tight view of it. See, there's a scale worm living in there. There's something else. I'm not sure what's going on with that polyp right there.
Yeah,
it's kind of inflated.
Going on.
Yeah.
You have a parasite of some type.
Play with the focus. The bright red thing, like this right here. Yeah, so that's a worm. It's a scale worm. You can see it's mostly tucked in behind, and that's one end of it. Yep.
Is this the one you've got on here as Dendrobathypathes?
Correct.
Question mark?
Yep.
Okay, I'll give it a click.
All right.
Okay.
Yeah, let me just check these two red colonies over here. There's a interesting looking crab here. This might be something different, Les.
Yeah.
Oh, it's a shrimp. I'm sorry. It's a shrimp that I'm looking at. Yeah, okay. And then there's a very small Anthomastus, just a couple of polyps there. Okay.
Very young one.
We're good to go. Center this up. Yep. Thanks.
Yeah, there's a question about the rock shape. Why does the rock look so much like new gravel boulders?
Yeah, let's have a look at that.
And
the question is, what's it growing on?
Yeah, this looks like a zoanthid colony.
Yeah, but is it on the rock? It looks like it. That's different.
Yeah, I think the issue about the rock shape is that this whole structure is extremely old. Like as we've been saying, 80 to 90 million years old. And there's been a lot of erosion over the years, and so most of the rocks, unless there's something that creates a new break or crack or something like that, most of the rocks are going to be rounded, and they're coated with a manganese oxide, which makes them look shiny. Manganese oxide is dissolving out of the seawater, or precipitating, sorry, out of the seawater onto the surface of the rock.
So here we're looking at a different kind of cnidarian. This is called a zoanthid. It's a colonial anemone. And you can see that this one is membranous. It's like there's this large mat that is growing over the rock, and then individual polyps are arising from that mat. In just a second, I'll get a closer look at that and show you. But individual polyps, so each one of those things that looks like a flower is a separate polyp. It's an animal. It has a mouth in the center surrounded by tentacles, and those tentacles, whoops, sorry Jay, that's why I shouldn't have zoomed in. I knew what you were doing and I couldn't help myself. Yeah. So yeah, a ring of tentacles surrounding the mouth. They have stinging cells, so they're related to sea anemones and corals. And we've seen several times, you've heard us refer to this glass sponge that has zoanthids in it, and so they're related to these ones, probably a different species. But you can see the way it's got that mat, that's tissue that interconnects the different polyps. And they can overgrow sponges and corals and all kinds of things. And one of the things we're still learning is, are there specific species that, for example, can only live on rock, or could this one also overgrow a coral or a sponge and so on? But this is the first time we've seen this.
It's the first time we've seen one on open rock.
Yep.
Absolutely. Yep.
That would be a sea star that I'm not looking at right now. Okay, so that's a nice little map.
I would say though that in the Atlantic we did see a zoanthid growing on open rock, but it was in the group that has the sand encrusted body wall, or it was on dead coral. It wasn't on a living coral or anything like that. But this is the first time I'm pretty sure that we've seen it just growing on open rock like that. That's pretty amazing, actually. It's a beautiful colony.
Another good indication of all these small white dots that we think may be forams, just how abundant they are. What's this white thing over there? Probably a sponge. That's okay. A sea star, another sponge.
You want to go into that one door?
Yeah, I think I know what it is, but let's pass it. It's the Walteria again, so we don't need to stop by that. Thank you. Yeah, even this is fine. We've got several zooms of that already today.
Yeah.
A couple
Are
more
we near
sponges.
the top of this knoll?
We're at 1463 and the top is about 1350, so we still got about 100
okay,
meters
so we
to
still
go.
got a little ways to go.
Yep.
Okay.
What are these small colonies nesting?
Where are you looking?
Just below her down.
Okay. Several fish in the area. Was it this sort of brownish thing?
Yeah.
Yeah, right there.
Yeah.
Yeah, I think that's something that's overgrowing a sponge. There's a cup coral next to it. Yeah, it's zoanthids. So this is another kind of zoanthid. So this is instructive. So we were just looking at that yellow mat, and we said it was a colonial anemone, a zoanthid. So this is a different species of zoanthid, and you can see it's overgrowing a coral. The white polyps that you see there are the primnoid octocoral, and otherwise this whole colony has been overgrown by this zoanthid. And
This is the one with the sandy grains in the body wall.
Yep.
You see all the little white spots all around the body wall of the
Yeah.
polyp.
look like gold
Yeah.
flecks.
Yeah.
Yeah, so they're able to incorporate things from the environment into the tissue. All right, so just have a look at this cup coral since we're here. So this would be a stony coral, scleractinian, and it's just got one polyp, the entire individual.
Oh,
so
yeah.
we refer to this as a solitary.
Cup coral. I got cup coral
Solitary
on
cup
my list
coral,
already,
yeah.
I think. Yeah.
Okay. There you go. And we'll see if that view means anything to the specialists out there. Thank you.
And you have a sea star, I think that's quite that.
On the, yep, there's sea star on the left. I'm not going to stop and look at it, but thank you. Tino is noting that the zoanthids may be able to use the scales that the corals produce as being incorporated into their body tissue.
It's an interesting idea.
Yeah. I know they can use sponge
Yeah,
spicules.
You could because you can consider the zoanthids to be parasites.
Absolutely.
So, and that's something a parasite might do, in fact. Eat the tissue, use the hard parts. Oh, here's more cracks with the brittle stars in them. We saw this yesterday.
Yeah.
Same kind of thing.
Maybe we can get a look at this sea star as we're coming up on it, because now we're in a position
Oh,
to
yeah.
see it ahead of time.
Over there that you don't want to see more.
Alrighty. It's your prediction? Oh yeah. Interesting.
So the area is not very...
So this, I'm not sure, is this Henricia like, Sarah? It's my sort of go-to for these long-armed ones. Zoroaster?
And there's a bat star.
Wow, look at this is all hydrozoan, I think, coating the bottom here.
Oh, yeah.
want to sit down here?
No, I don't. I want to go a little bit further where the discoloration on the left was. I see some solid sea cucumbers down there. Yeah, let's check out that rock. We'll sit there. That might be a good spot to see this. So what comes first? All this hydrozoan and stuff growing on the bottom, and then that attracts the urchins. We're seeing a few urchins here and there. And yesterday they were absolutely covering the bottom. And we didn't see all of this feathery brown stuff that you see, these hydrozoans. So perhaps they're grazing on them. And there's one of those sea stars, Les, that's a pentagonal disc with the short arms.
Oh yeah, the short arm brittle star. Yeah. Pentagon brittle star, I'm gonna call that.
There was a question about the nitrogen and oxygen levels of this area relative to other parts of the ocean.
Our oxygen is
hydroids everywhere.
7.6 micromoles.
So I don't know, could those be like little amphipod tubes? I'm not sure that these are hydrozoans, actually.
Huh. Well, they could be forams as well.
Is that a nudibranch right there?
Hmm?
Is that a nudibranch?
Where? Oh, right there. Oh, yes. Well, there you are. Yeah, sure looks like it.
I don't know.
Yeah, I think it is.
Yeah. I should stick with my first guess. Is that what you're saying? Yeah, it is.
Huh.
So that's a little
That's the first time we've ever
nudibranch.
seen a nudibranch.
Yeah, especially like that. So this is a kind
Wow,
of
that's
snail,
amazing.
basically. It doesn't have a shell. And what you're looking at, those little inflated sacs with the white tips, are extensions from the dorsal surface, its back. At the lower side, lower left, you can see two long tentacles. And the white stuff in there are called cnidosacs. These are mollusks that are able to feed on cnidarians, steal their stinging cells without firing
There's
them,
another one up
transport
there.
them into its
There's another one up in the
gut.
top left.
Yep. we'll
So
look
I
at
think
that
they
one.
must be associated with these hydroids or whatever these things are.
Yeah, that gives me
See,
more
that one's
confidence
eating.
they're hydroids.
Huh?
That gives me more confidence that these are hydroids.
Yeah.
So yeah, they're able to feed on the stinging cells but not fire them. And
Yeah, that's
second.
definitely a hydroid.
Yep. And then transport them to the
Huh.
tips of these extensions of the gut
Yeah, look at
so that
that.
they protect themselves.
Huh. And there's
Very
also
cool.
a very small snail right in the middle here. There,
Yep.
right there.
Yep.
There's
Actually,
a
wait,
lot
is
of
that an amphipod?
very
That's an
tiny
amphipod.
things. What's that?
That's an amphipod.
Oh, that's an amphipod.
Yeah.
Ah, yes. Now I see. Yep. Oh, yeah.
Well, that's really cool.
Huh.
Those nudibranchs.
Some of these forams are up on the hydroids.
Those look like snails, Les. Those
Well,
are snails.
That's what forams do look like. Snails. They're coiled.
Oh, I agree, but those
Yeah.
those have a gut.
Yeah.
Forams don't.
So they're awful small. I mean,
They
one
are.
of the problems with this lens is that we're looking at it like we're looking at it through a microscope.
That's right. But the cool thing is you see all of this brown
Yeah.
fuzz and all these rocks, and then when we go in really close, you see that there are a variety of organisms that are
Yeah.
taking advantage of it as food. All right. Wow. So you can just get a good sense of how much is out there. Look at the fuzz
Yeah,
in that
there's
rock.
fuzz. Look at the fuzz over on the right-hand side as well. I think these are definitely growing like in a hydroid pattern. They may be not. They may not be currently alive, but there's something. Then there's something growing on the chitinous material left behind when the hydroid has died. I think that's a good possibility.
Certainly alive enough
Yeah.
to sustain these snails, the nudibranchs, and maybe these urchins as well.
Yeah,
All right, we'll
yeah.
move on. Good stuff.
Well, it's a little micro zoo.
Yeah. Absolutely. That's right. Micro
Yeah.
menagerie.
Yeah, I mean, this is the thing that that
Let's
you rarely,
look
I
at
mean,
that right there, please.
yeah. People are always fixated on the larger things like the corals, but in fact, the seamount's probably loaded and covered with all of this small stuff. It's just that you don't get a chance very often to have a look at it.
It's nice when you have a deep sea diving microscope.
Yes, that's right. Yeah, not too many people have the opportunity to see the video through the lens like this.
We're gonna check out and see. This looks like maybe we have a Walteria sponge overgrown with hydroids. We'll just verify that that's what this is.
But now through the wonders of the internet, and people from all over can have a look at exactly
Absolutely.
the same things that we're seeing. There's another chitin up there, but it's got a red dot on the end of it.
And
I don't
is
know
this
what that's about.
there's a crushed shell down there.
Oh yeah, there's a broken shell of some type.
Must be the urchin. No, that's okay. I know what this is. So that's basically a dead sponge being overgrown with the same kind of hydroids that are overgrowing all this rock. And then I think that white thing you saw was a sea urchin dead. That's the skeleton or the test. Let's check out. Is this direction okay, J-Rod, or is that too far out? Yeah.
Tina was commenting that the hydroids do not look very healthy.
Yeah. It's unclear to me whether they're healthy or not. Okay, that's that Lefroyella sponge. We've seen that several times, and you can kind of see the orange dots in it. Those are anemones or zoanthids that are growing in it.
You should get a good look at the surface of that bat star over there when we get a chance also.
There you go. So these are
very
actually
nice.
anemones that are growing the sponge, not zoanthids. So each of these is individual. They're not connected to one another. There's some amphipods. All right, we collected one of those sponges yesterday, I believe. And we're going to look at the sea star that's on the left, the white one. Sea star is a member of the class Asteroida and the phylum Echinodermata, and just above it you see a brittle star. It's got a much smaller disc with long, slender arms. That's a different class of Echinoderm, the Ophiuroida, and then you see the chitin in between them.
Yeah, get a nice shot of that. We could get it identified, I think.
This looks like is this porcelain asterid?
It's a goniasterid.
Goniasterid.
What is it?
Goniasterid.
But it looks, there is
Oh, a goniasterid. Oh, yeah. Okay, I'll change that on here.
Yep, and there's a snail on the left over there. All right, I'm going to zoom in here.
Yeah. Oh, that's nice. Can you get a zoom in a bit more on that?
I'm just trying to do it in stages so it's nice and stable.
Okay. Yeah, that's good. Send that to Chrisma.
Yep.
The plates are not completely contiguous in the center part of that.
No, they're not.
Either.
I think they're basically like spines raised on little pedestals.
Yeah.
So they kind of interlock together. It's not a.
Yeah, right.
smooth
That's
disc.
I see. Yep. Nice.
And we'll just pan over
There's
here.
the
to
chitin
the
that we've
left
seen
and
before.
See that, the snail's kind
The
of stuck in
snail.
there.
And there was something else a little bit above that looked.
No, that's the solid less.
Oh, that's what it is. The solid. Yeah.
that's
It's
sea
a very
cucumber.
tiny solid.
Yeah.
Yeah.
No, that's okay. Yeah, J-Rod.
Yeah. That's what it was. Yeah.
Okay. Continue our trip.
What's the name of that sea star?
The name of the sea star was suggested to be Goniaster.
There's something up there in top left, it might just be urchins.
Let's check it out. Yesterday the urchins led us to the Paragorgia, the bubblegum corals. I'm wondering if we're going to see those
Well,
again
we're
at
in
the
the
top.
yeah, we're in the depth range. Yeah.
There's another sea star, I think, that we're passing over. The lasers are just passing over now.
Now here's
Yeah.
a rock promontory with urchins on it. This is what we saw yesterday on Yomie Seamount, the same depth.
It's kind of interesting topography, isn't it?
Yeah.
Not as many urchins as we saw yesterday. Boy, it's really broken up and jumbled.
Not as stable.
Not as stable at some point in the past. Larger one of those Lefroyella like sponges that had all the anemones growing in it down there.
Look at the layering in the rocks.
Yeah.
The rock has layers. It's like it's sedimentary.
Yeah, right in the center here. Yep.
Yeah. This is a sedimentary rock. Look at that. It's like a sandstone. Can we go down and look at the
Sure.
one down in the lower left or lower right here?
Yeah, just where the urchins.
Take a
look right
nice
here.
close look at that. Wow. Huh. So that was probably above sea level at one
Yeah,
time.
These are probably lagoonal
Yeah.
deposits or shoreline stuff.
John, you want to come in and describe that a little louder? John Smith here from the University of Hawaii, and you can maybe tell us a bit about the geology.
Yeah, so these layered ones would be sedimentary rocks, and depending what they're made of, they're either probably shoreline sand, or if they're more fine grained, they'd be like a mud or a silt, like a lagoonal deposit. So so hard to tell from here, but you can see the layering.
Would this be like when you see the changes in the tilt of the layering? That's like wind blowing. Would that be more like
Well-
wind blowing sand?
Yeah, I mean, you could have dune preserve, but you can't really tell from here. All this could have been tilted and
Yeah.
readjusted, earthquakes and
Wonder if we
settling
could get
of
a piece.
the
Well, there's,
subsidence.
there's one other cool thing I'm seeing. I'm going to zoom in here because it looks like you can see the way this is cut on the edge. What we keep talking about, the layering.
Yeah.
It's, excuse me, not the layering, the crust that is formed.
Yeah.
So look at the edge right there in the center of the screen,
Yeah.
where the layers end. That looks to me like right there,
Right
broken
there.
oak,
Yeah.
Yeah.
broken
Actually,
crust.
Another explanation is this could be volcanic ash fall, and it could be submarine
Oh
generated
yeah.
too.
Yeah. It
That's-
could be an ash fall.
Well,
That's
it's hard
true.
to tell with the algae and without getting a sample of
Yeah.
it. So it could
Well, that's what I was gonna just wonder whether we should see if we could
volcaniclastic material. Yeah.
find a piece of it that we could pick up.
So that might actually be a better
Maybe there is
explanation.
some laying down at that. There's a whole
Yeah, if we
area
could get
here.
a piece of
Look
it.
at that.
What about like right there?
Yeah, like that one right there. Yeah, and there's one above it also that might be possible. To
Yeah,
get
it's
that.
actually looking kind of more like volcaniclastic stuff, but
It's looking more like what?
I don't know. I mean, it looks kind of like could be ash deposits.
Uh huh.
See if one of these is loose.
John?
Yeah.
Yeah, it might be kind of crumbly because it is made up of loose
If you
material.
want to sample one here, right, I can just fly around and find another little sample
Yeah,
size.
something bigger than that, than a piece like that. Yeah.
Well, even if you got what was left of that, that would be fine.
You got it with the cameras and stuff?
Yes.
Enough said.
Ever see anything down here that you like? You want something that has a like a striation, right?
Yeah, the layering. Kind of just depends what you can grab. That's there's this piece. That looks pretty good, but I don't know if you can get it.
Big triangle?
Yeah, the wedge with the crust on it, and it's kind of a nice.
Talking about, right? Triangle at the bottom of the screen.
A rock pie.
don't come off in small chunks apparently.
Yeah, it looks like we had some kind of a collapse here that exposed the whole outcrop, which is fortunate for us.
Can we drop it in Rockbox 2?
2?
2, is that alright with you? Or is that...
We could try. I'm just worried about his.
Okay, go
grip.
one. Go one. Go one.
Yeah, it's pretty
So I'm just gonna
obvious.
start forging out and hammer back. You can just keep coming in.
I only said 2 because I wasn't sure the size relative to the rock that's already in there.
You're gonna have to warm up a little bit. Bring the forge out a little bit more. Or are you coming in? Now that we're safe right here, hold what you got. Just free. I'm just going to forge out. Nice and easy. We might be able to get you in the rock box too. Now that we're in a safe spot.
It was so big, and there's already a big one in there. I was concerned. But...
That one got layers.
Yes, it had layers. We just kind of rotated it, so it might not be obvious now. Yeah. It also looks like it has a stoloniferous
arm up a little bit.
coral on it.
I mean,
Yeah, it can
you might
go-
be able to bat it around too. So yeah, I got more porch out. So just hold it a bit. Porch out. There you go. Just be fully porched out. So yeah, we just have kind of just don't shoulder up too much more.
John, there's a question for you. Could this have been part of a pyroclastic flow?
Yeah, it's possible. This could have been a volcanic pyroclastic-like flow, or a series of
Like I said, that's a
layers
big rock.
from a series of events. Hard to tell until we get it aboard.
Rockbox 2, then.
These layered
A
deposits
wonderful
Nope.
That's excellent.
look similar when you're
one. Sample number?
looking at them like
Oh,
this.
sorry, yes, 104-1. Sorry. What's that?
Just so unusual to see a layered rock on any of these seamounts. That's what I thought.
Yeah, we've seen this up in the northwestern Hawaiian Islands in the monument there in Papahānaumoku.
Is that right?
On the, some of the shallower dives, they look,
Yeah.
they're banded for sure, and a lot of them turn out to be lagoonal sedimentary rocks. But we know from Yomie Seamount off the Big Island that you can have submarine volcaniclastic rocks that are in layered.
It looks like this thing is continuously falling apart. You can see where the coat is
Yeah.
broken away from the coating.
Yeah, so if it was coated with the manganese was intact, we probably would have gone right by it. Never known it was any different.
A lot of times
Now you can kind of start looking at this,
it
and
means it's going to be a
now that you kind of know, you can maybe pick up some hints. But
Right, sure. It makes like a talus slope below it.
yeah, talus, that's the root.
I share your optimism, J-Rock.
That's outstanding. Just rock, yeah.
yeah, good grab.
Well, I'm a sedimentary type guy, though. I started working in the mud.
Yeah.
I didn't start with all this volcanic stuff.
Are you a sedimental guy?
Yeah, right.
Oh my gosh. Here they come. Here they come. Oh, what's this? Is this an anemone?
Old geology jokes.
Winding them up here.
Not a bad joke.
So this is an anemone we saw yesterday with these sort of furrowed
yeah.
edges.
Oh, wait a minute. Why
Well,
is
maybe
that
not.
white outer edge?
Yesterday, the one we saw, we thought maybe was Isoactinurus.
Oh, it's the
I'm not
brownish.
sure if this is exactly the same
It's
thing,
dying.
but definitely an anemone.
Okay, I gotta record
It's
this.
partly retracted. Alrighty, that's good. These are really difficult to identify if you haven't collected any.
Okay,
It's not
so
something we're
we're gonna.
collecting.
We have a light possibly on the almost the middle of the screen now.
See the white one?
Yeah, let's check that out, please.
And another one further back.
I said right, I meant left. Fortunately. Yeah, there's another primnode. It's the same primnode. Maybe by the end of the dive
Yeah.
we'll collect one, but I do think it's what we got yesterday. On the right, tucked down in there is a hydrocoral. Another chitin, a few brittle stars. That's good. We can go.
And you have another one up ahead, slightly closer right.
Yep, another primnode, little white bush.
In response to the question about having a sample of the rounded rock, the answer is yes. We've got several samples now that we've taken both on Yomie Seamount and one here today on this one.
See more of these branch sponges on the right that are overgrown with the hydrozoans. There's a yellow feather star there. Lefroyella sponge.
Thank you, I see it up to the northeast. I'll bring us over.
Alright, I'm just going to tilt the camera just to make sure I'm oriented the way I think I am. Okay.
Another primnoid to the left.
Primnoid on the left, yep. Sponges on the right. Sponges in the distance on the right. Walteria type. I mean on the left, sorry. Okay. The current moving from the left of the screen to the right of the screen. Our heading right now is north. So that would suggest it's coming from west to east. We're at 1,457 meters depth here on Yomie Seamount. We're on the north end of the seamount. We're at 42 degrees, 26 minutes north latitude. Yep, dead sponge, fallen over. The red mushroom coral Anthomastus. That's the stalk of a sponge. Also doesn't look in very good condition.
There's a question again about these seamounts being islands in the past.
Yeah, this Les was discussing some evidence from deep sea drilling that suggested that in the distant past, this seamount had risen high enough that it was exposed above sea level, so it was an island, much like Hawaii is today.
Right.
And there were plants growing on it.
Yeah, in fact, in the drilling borehole on Suiko, here comes a big
Macrurid? Yeah. A grenadier? Is that
the Pacific Grenadier, one of the big ones. But anyway, the borehole on Suiko Seamount produced pollen from about the Cretaceous age, and the pollen represented maidenhair ferns and other families of ferns, as well as tree ferns. So there weren't like palm trees and coconuts and things like that, but it was definitely a tropical island and had a typical tropical lagoon in the center.
It's another Anthomastus colony, but is this different from the one that we were imaging earlier? Is this Anthomastus different from the one that we were imaging earlier, or is this the same?
Yeah, we need to look at the side, the stalk on this one. I think this might be the same as the one we collected.
Okay.
Yeah. Oh, yeah. Yeah, looks like it. Whoops. There you go. Nice image. Yeah, really nice. Oh, look at this polyp on the lower left. Looks like it's starting to get eggs along the mesenteries there.
On it too.
Huh. Ah, nice. Yeah, yeah.
You can see all the little siphonozoids, those little white bumps on the surface. Those are a specialized form of polyp. They don't have the tentacles. They're obviously not large like you see these other ones. One of the things that's really nice about this is you can see the classic hallmark of an octocoral is that it has 8 tentacles surrounding the mouth, and those tentacles are what we call pinnate. It's those little side branches that you see. And these polyps are so large, it's very easy to see, but all the octocorals we see have this same structure to them. All right, that's rather nice.
That's red. I've got that one already.
Yes, thank you. Cup corals.
There are cup corals here, but they seem to be really isolated one from the other. Not
Yeah, I
very
agree.
many.
On the first dive on Suica, we came to an area that was pretty steep, and we saw lots of cup corals growing together.
Yeah.
But since that time, we really haven't.
No, we haven't
to an
seen anything
environment
like
like
that
that.
since. Exactly. Yeah.
Sponges, couple of urchins. You know,
But
we
that
pa-
was also deeper. And so maybe tomorrow's deep dive will have something similar.
Looks like there's maybe a little stalk right here.
Oh, yeah. But I'm getting the sense from the spreadsheet here that the shallower dives have some of the same fauna as the deeper dives, but the shallow dive from Yomie Seamount and this seamount look more similar to each other than they do to the deeper dives on the same seamount.
Is it possible to get around behind this? This is a black coral. I just want to see the branching structure to see if it's the same or if... Yeah, at first I thought it might be Bathypathes, but it's not, because those side branches that come out of the main stem are also branched. And
This is
so
a
this is
dendrobrad.
probably, yeah, exactly. It's
Yeah.
just a small colony of a Dendropathes. That's good, J-Rod. I got what I need. Thank you. Shrimp down there? No, you're fine. You're fine. Keep going. Yep. Can't say I got what I need and then start looking at it. It's like, wait, what did you say?
Oh, who do we have here? Our big grenadier.
Haven't had any real good close-ups of this guy, so.
You could zoom in on the head and that dorsal fin, and then maybe we can get a confirmed identification on this thing.
See, it's got that extra spine on the dorsal fin.
Yeah.
Rat tail.
I see, and it
Yep.
looks like there might be
Rat
a
tail
barbell.
here is the same. There's multiple.
There's a barbell under there where the mouth is. And I don't know if there's anything significant about the nostrils. But
Nice
yeah.
view. Should be able to count the scales there.
yeah.
Yeah, I got lots of screenshots.
Okay.
Yeah, those eyes is right. But you're right, it doesn't seem to be any parasites at all, interestingly.
What part do you want to see?
I got it. I got one.
You got it?
Yeah. Thank you. All right.
I wonder if he's eating the little star arms.
I'm going to predict sponge, but I'm not 100% certain yet.
The Anthomastus on the left.
Anthomastus, yeah. Or unless it's another zoanthid, Matt.
Looks like we've flattened out a little bit again. Like we keep going up steps.
Yeah.
That's fine.
I think dead sponge is a good guess. Turned over a rock. Yeah, it's a rock. It's the underside of rock, just like this one that has the crab on it.
Yeah.
Huh.
And you have a sea star on the top too.
Oh yeah. Sea star up here. There's a cup coral right just to the left of the lasers.
Cup coral, chitin, brachiopods, all the same guys.
Yeah. Like you said before, you know, there's lots of small stuff when you get down to it. There's a little bryozoan colony also there. Let's see if I can get on this guy. There you can see those pedestal-like spines on the arm of the sea star.
Looks like look actually.
No, that's okay. I just wasn't sure if somebody else was.
Oh yeah, that looks like a bryozoan.
Yeah, so that nice side view, you can see what I mean. Unless you had said earlier, it didn't look like those plates were connected. You can see they
Oh yeah.
stand up like little spines. There we go. Bryozoan in the background. Okay.
Hmm.
1,459. Flat. No bubblegum corals yet.
No. We should have seen them by now.
Another one of those
Across them.
red brown anemones on the left and an Lephoraella sponge in the center.
Maybe when we get right up onto the ridge,
Yeah.
there'll be more flow, and then we'll get them.
Yeah, I thought, you know, the overall setting, we'd have enough flow moving across the seamount
Yeah.
that this whole ridge would be bathed, but maybe we need to get right up on top.
Yeah, I think so.
So,
I don't know how much we're pretty close though. We must be getting
If you
pretty
want
close
to
to the
let
top.
them know, the overlay that they have
okay.
shows a depth that's 20 meters off. It's a depth that's not as calibrated as what we're using in here.
Okay, that 1484 you mean?
Yeah.
Oh,
okay.
Yomie
Yeah,
Seamount.
It's not the CTD, that's
Okay, so the video screen that you're seeing on shore there has some other information like our heading and our depth and so on. And just so you know, the calibration of the depth that you're seeing on the screen is about 20 meters different from what we are seeing in here. So we're actually at about 1,460 meters depth. Now you see an inset that shows where we are out in the Pacific Ocean. Pretty remote. We're in the far western Pacific Ocean. We're actually crossed over the date line. So depending where you're watching from, maybe different. We're zooming in here on the Emperor Seamounts. This is that chain that was running from north to south, and there we are on Yomie Seamount on the north side, kind of on a plateau. That's a multi-beam bathymetry map you're looking at, so that the color is associated with depth. Were you calling me numbers?
There's a crab on the, well,
Two crabs, yep. We'll check out these two crabs. This is the Macroregno.
Two of them.
Macroregnonia. Thank you, Sarah. Sarah Bingo on the back there from University of Hawaii, who's been annotating video from the Pacific for a few years and is really probably more familiar than anybody with the Central Pacific fauna on the video. Yes, I see the coral. I think it's a black coral. Thank you, crabs, for leading me to this coral. And if you don't mind, I'm going to skip the crabs just for a second, just to get a quick view and see what that is. Yeah, that's a very small baby black coral. So now we can check out what these two crabs are doing. One of them's missing a leg. Yep. Follow me, follow me.
Well, I wouldn't be surprised if one's a male and one's a female.
Yeah. One of the things that's interesting, for the viewers, the only reason we can see this is because the lights that are being carried by the ROVs. So normally this would be completely black. We're well below the level at which sunlight can penetrate. So these crabs probably can't see each other. So they're sensing each other in another way.
I can guarantee they can smell each other.
Absolutely.
How many lumens do you think you put out?
The ROV?
Mm-hmm.
Oh, there's a question on shore for you. How many lumens can the SuBastian put out? We'll give you a couple of minutes to come up with some guesses. Yeah. That's right. Call a buddy.
Yeah.
All right, I think
So,
that's good for the
One thing
crabs.
about when you have a pair of crabs like this, especially when the female is getting ready to molt, usually the female produces a pheromone that is attractive to the male, and the male often then will go into what's called a mate guarding behavior, and that's because the female, in order to lay eggs, has to molt, and so the chitin is soft, and then the eggs can be extruded. And so she's vulnerable at that time to predation, and so the male guards the female until the cuticle of the female hardens again.
Yeah, if they would be cooperative and tilt upwards, we might be able to tell you if it's a male or a female by looking at the way the shape of the abdomen that's folded underneath there, but can't from this angle, or I can't anyway.
No,
Nothing.
and there's just a little shrimp or
Yeah,
whatever.
this little shrimp crawling underneath, taking a chance.
Not interested in being crab food.
Yep. These guys look like they're picking at the substrate.
They could be, in fact, they're sort of like a spoon-shaped structure on the fixed finger of the claw, and maybe that's in fact what they're doing.
Zoom into the
You
mouth
great.
parts of this one here.
You want to make sure you don't get too anthropomorphic about what's going on here with the female looking like she's feeding the male, but probably trying to figure out who's where he is. Yeah. These crabs look like they do have small eyes on the stalks are reduced and the eyes are reduced, and they're probably close to blind. Might pick up some bioluminescence or something, but that would be it, I think.
let them do their thing.
Interesting. Well, we don't get to see this kind of behavior too often. Usually we see these things one at a time.
If you pan up, there was a coral right there.
Yeah, I think that was the same primnoid again. Maybe I'll get a quick look, just to not make too much of an assumption. It's got something on it. Why is it purple in the middle?
Yeah, exactly.
What is going on there? Is it just shadow?
It's a pretty small colony. It must be the shadow.
Yeah. Yeah, that's that same primnode. Yep, this shadow. Okay. That's basically the only primnode we've been seeing today. In terms of octocorals, I think we've seen Anthomastus and that. Have we seen anything else?
Yeah.
I think that's all.
What's
Right?
that?
I think we saw the bamboo coral, of course. We collected one bamboo coral. We saw a second colony, but in terms of octocorals, we've seen those 2 bamboo whips, several of these primnode fans, and Anthomastus, and I don't think we've seen anything else.
Yeah.
We've seen the black coral, Dendrobathopathes. We've seen some hydrocoral. We've seen some stoloniferous octocoral.
Yeah, but actually the coral diversity is pretty low,
Yep.
all things considered.
Maybe 3 species of sponges. These left royal, like on the top left, there's one. We've seen, I think we saw at least one vase type sponge. We've seen the Walteria, of course, the branching Walteria. No, that's okay. I think, Les and I are curious about what the top of this ridge is going to look like, so I think for the most part, yeah, because we're not seeing anything different right now. No doubt as soon as you put on the gas, but for now. Sound right?
Yep.
So you're tuned into live video from the Schmidt Ocean Institute's RV Falkor, and the video is being transmitted from the remotely operated vehicle SuBastian, which is diving right now 1,461 meters depth on Yomie Seamount. This is a seamount at the northern end of the Emperor Seamounts. We're in the western North Pacific, and we're studying the deep sea coral and sponge communities, trying to address biogeographic questions to look for where do you get a transition from the communities you see in the north to the communities you see in the south, and then relate that to water column characteristics, the chemistry, the nutrients, the current flow, and so on. So we're on this expedition for about 32 days. We've been out here for about 14 days now, and we'll be hopefully diving for the next 8 or 9 days.
Tiny whip, I'm not sure if it's a coral
Oh.
or if it's something else.
Where?
At the bottom of
Oh
the
yeah,
size.
that's a Walteria sponge. Thank you. Yep.
Yeah, it's good.
Good eye, Natalie. So as I say, we're on Yomie Seamount. We've had a couple of dives on Suiko Seamount, which is a very large seamount to the north of us. And we did one dive from about 2.2 kilometers depth up to about 1,700 meters depth. And then we did a second one at 1,500 meters to about 1,350 meters. And the dive we're doing today kind of replicates that same depth interval. So when we first got down to the bottom, it was 1,520 meters depth, and now we're at 1,461 on our way to the top of this ridge feature that we're climbing. And our heading, we're almost traveling due north, and this ridge, this elongated ridge, which is basically a ridge on the top of the seamount, is oriented more or less east-west. So we're climbing the wall of it, and then when we get to the top of it, we'll turn and follow it to the west. Let's have a look at the star that's coming in the top left. I think this is one of the sun stars. We saw several of these yesterday, but we haven't got a good look at it today. I do not think we have a Geiger counter.
We have one on board.
Sorry, I meant on the ROV. But we do have one on board, so yeah, I guess we could potentially look at the rock. I think I'm going to want to look at this afterwards, J-Rod. Thanks. So I keep calling this sun star. It's Pycno something. Sarah, do you know?
Solaster.
Solaster, and what's the larger group that's part of?
Solastaridae.
Solastaridae. So the family Solastaridae, maybe the genus Solaster. And you can see it's a sun star or multi-armed sea star. Okay. And then I noticed there was something kind of thin and white branching. We're going to go over and take a look at this and see if this might be a different kind of coral. Still don't recognize what it is, which is a good sign.
Ooh, the Crustacean.
I'm intrigued. Whatever it is, I'm intrigued. It might be just a scrawny primnoid, actually.
Yeah, right.
Afraid so. Oh well. I was hopeful there for a moment.
I think we better collect
Yeah.
this.
You don't think it's the same thing? Just in worse shape?
Wow, look at that. Look at those little tiny polyps. Huh. I think it's a primnoid, but I don't, unless it's that little Arthrogorgia that we got, it's something else.
Really?
Yeah. Oh, yeah. Look at the size. Look at those little polyps. Oh, yeah, we got to get that thing. Is that on a rock?
It is on a sponge.
Oh, it's on a sponge?
Well,
You could go in there and
Yeah.
grab that whole bottom
Yeah.
and pull the whole thing up.
right.
It's not very big, right? So
No, it's pretty small. Worse than lasers.
we should take the whole thing.
So that colony can't be more than 15 centimeters tall.
It's how much?
Maybe 15 centimeters.
Oh, okay.
Did
So
you
that
copy
would
that,
be,
Jeremy?
yeah.
Yes, obviously you did. Yes, we are. And what
Yeah.
we'd like to try to do, if you can, is this is a sponge down here, and it appears to be growing off the sponge.
Right.
So maybe if you just grab the sponge, you'll get that whole
I'm
column.
just gonna change my
Yeah.
setup for that.
Okay.
I'd put it in a bio box if it's 15 centimeters.
Yes, we can go in a quad.
That's the possible.
Say again?
I was gonna say, if it,
What's that now?
There's another little like white ball, maybe a corallid or something. What's wrong?
Yeah, I see what you mean. This will be 105 one.
Try to get you close, but not too close. All right.
It looked like it had too many scales for Arthrogorgia.
Huh?
I think it has too many scales for Arthrogorgia.
for a Crustacean?
Arthrogorgia.
Oh, Arthrogorgia.
It's
Yes.
too many scales.
Yeah. It's something really different.
Amy, I see your comment. And as you can see, we are collecting this primnoid, and I agree with you. I had said earlier that if we weren't going to be finding a lot of stuff to collect, that we would certainly collect this primnoid that's been fairly common throughout the dive.
He wants the coral that's possibly attached to the sponge, so he wants to get as close to the rock as possible. That's correct, right?
Yeah, it's the white coral that's of interest, and I just thought it would be easier to grab that round sponge that we did.
Got a view on 6 feet away, trying to move it in for you. What's that?
I think you're going to have to go a little bit further because the disc that's holding it to the sponge, you haven't got your jaws around yet. It looks like the whole sponge is coming loose, so that's good. That's good. Yeah, just close down now.
Pretty nice.
Yeah, yeah, that's good. It's
Got a camera in. And BioBox 1.
BioBox 1, correct? Yep. The nearest opening to wherever your claw is.
You on that? Okay. Hold it again. I'm going to go ahead and open the BioBox 1 nice and easy.
Anyone.
Anyone you want.
They're all.
Watch out.
Yeah, nice, 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 nice.
Nice collection. BioBox 1B YOM 1051. So I noted that also had a brittle star and a sponge. Yeah, that was on a convenient substrate for us.
Good to go.
Just if we could look down, or if I could look down. Before we go, there's one stalk. Yeah, that thing. I want to look at it, the white thing in the lower left. I think it's a hydrocoral.
Mm.
It's one of the larger colonies that we've seen today, though. Yep.
Okay, yeah.
Okay.
That
Thank
was
you.
good.
Yeah, we're good to go. Yeah, that was a good collection.
That's a nice colony.
Anthomastus here, the red mushroom coral on the right. You know, for the most part, the corals, even the sponges, but especially the corals, ones we do see, they're all keeping their heads down. You know, they're all very short, low, and
Yeah,
not
exactly.
sticking up very much into the water column.
Yeah, I don't understand that,
Yeah.
but that's right. I mean, nothing's, except for the Paragorgia, nothing's been tall.
Correct. So this is not what I would call, you know, a coral forest or a coral meadow.
No.
It's,
No.
I don't know what the name is we come up with this, but
Yeah,
underground corals.
everything's hyper dispersed.
Yeah.
Yeah, I don't know. It's kind of interesting because, you know, as we were discussing yesterday, there's plenty of food.
Yep.
It's not, food is not the issue. I mean, there's lots of food. So, I don't know what the, it must be larval delivery. But again, why are things so short? I mean, once you get here, you would think you could grow for 100 years and
Sure.
get big, you know?
Today we can't say that it's an urchin barren, right? We're not
No,
seeing
there's
urchin
no
barren.
urchin barren today.
Yesterday we saw literally thousands of sea urchins which would be
Yeah.
grazing over the substrate, so any larva that settled would be at the risk of being eaten fairly quickly. It's not the case here.
Could it be
Yeah.
that there's a big colony up current of them?
We've seen sort of like a half a dozen.
Sure.
Yellow colony that too.
Side of the seamount gets
Yeah, so definitely. So both of these comments our ROV pilots are asking about. The first was, imagine that there's a really big colony that's kind of upstream of where we are and that's pre-filtering the food. So absolutely something like that could be happening, although there would have to be a lot of them and awful big to have this kind of an impact. But the other comment was, you know, maybe we're on the downstream side of this ridge or this seamount, and although we see this current moving past us, the dominant current is coming from somewhere else. And so upstream of here is where the big communities are. And absolutely, that is a possibility. It's one of the reasons why Les and I are kind of eager to get up to the top of this ridge, because maybe up there we're going to see all these corals and sponges aligned, catching the current that's coming over from north to south.
There's a different fish with his tail sticking out
Yeah, I
behind
see
the
that.
rock. Find out who that is if we
Is
can.
this some kind of hake or something? Sarah?
Yes.
Is this a hake or something? Hiding behind this rock?
It looks like a morid, that shaped tail.
Yeah.
Yeah, morid. Yeah, I think that's probably right.
I don't know, maybe the hake are more common in
If
the
I could
Atlantic.
see the dorsal fin, I'd be able to tell.
You have to see its face to know what kind of morid it is.
It's the shy kind.
Yeah.
But morids include cod as well.
Okay.
Yeah. And it's a blue entomora.
So it wasn't a terrible guess that I had then.
Yeah.
No.
Huh. Wow. Huh.
It's got quite a prominent spine on the dorsal fin there.
Yeah.
Yeah, that's probably a blue entomora if it's got a big spine. Yeah. Oh yeah, I see it. Yeah. Yeah. It's a globally distributed fish.
Nice
but,
lateral
but
line.
never in really big high numbers.
So like,
Alrighty. New fish for the day.
oh, there's a little.
It's a black coral stuck in a rock there. Tightly stuck in a rock.
Yeah,
Kind of a bizarre place to be growing, huh?
Go ahead and look at it.
I don't know. Is it? No, is that a Swiftia? Ah, slow down a second here. That's a Swiftia, I think.
Oh,
What are you doing in there?
Huh.
Absolutely.
Hiding from you?
That is definitely hiding from me.
Yeah.
Nice try.
Can you go in there and get it? That's the
I don't
question.
think so. I don't think we should try to get that.
Okay.
Look at where that is.
That's a
It's
Swiftia.
definitely worth noting. Yeah.
Yeah, I'll probably write them on the list, Joe.
That's a prostitute, yeah.
Yeah. So this is an octocoral. I thought it was a black coral from a
Pacific
distance
like
because
that.
of the color, Black corals are the order Antipatharia, Hexacoralia. This is an octocoral, which is the sort of dominant fauna that we're looking at down here. And this is the first time on this cruise that we've seen this genus of coral.
Yeah.
Interesting. And this is a genus Swiftia that is also known from both the Atlantic and the Pacific. There are a number of different species. And
Just when we were saying,
I guess it's a separate
delivery.
stoloniferan octocoral growing as a ribbon on the rock on the right side there. I don't imagine they're related, but wouldn't it be cool if this was arising from that?
Watch.
Is that net? Oh, the isopod feels you. There's a little isopod there that Les was pointing to, and every time he pointed to it, it would scuttle a little bit.
Yeah.
There you go.
Little
your,
Acyloidean isopod. I'm not can't quite get enough detail to know which family it's in, but
Basically a deep sea roly-poly.
looks like a
Munopsidae?
Huh?
Munopsidae? I think it is.
munopsid. Yeah, exactly. Yeah, that's what I was thinking of. Yeah.
What is this called?
It's Swiftia. Yep. So if we find one of these that's not tucked into a rock like that, we would like to collect it. But I'm not willing to spend time trying to wrestle that out of there. Yeah, that's the Macroregno?
No, this is. I think this is a different one.
Oh, okay.
Oh yeah. Yeah. So that isopod wasn't a roly-poly because it's not capable of folding up into a ball like
That's right.
the
I only
terrestrial ones, but
I only meant roly-polies are isopods, so for people who don't know what isopods are,
yeah,
we can...
roly-polies are isopods. But
Yep.
yeah. I have plenty in my wood pile at home.
Yeah.
Well, actually, I had them in my basement this year. Yeah, I don't know how they were getting in. They weren't getting out.
Swiftia. How about that?
Yeah. Well, that's one that I thought that we should get here.
Yeah.
So
Good
we
catch,
don't
Natalie.
have a corallid today either, do we?
Nope.
No.
No corallids, no paragorgians,
No Paragorgia.
certainly no chrysogorgias. I haven't seen one of those yet in this expedition.
Yeah. Yeah, lava lice.
Actually,
yeah,
that's not true. We saw one at Hess Rise.
Huh?
I said that's not true. We saw one at Hess Rise, a chrysogorgia.
Oh,
We
yes.
didn't collect it, but we saw
Right,
it.
that's correct. But that doesn't count.
That does not count. We're on the Emperor Seamounts and determining where the boundary is. I agree.
Yeah.
Lava lice. That's a good
Yeah,
one.
I like that. Lava
That
lice.
is good. Yes, these are lava lice. Emperor Yomie is a little bit itchy today.
Yeah.
There's a small coral,
Well, there's some
maybe primnoid,
kind of
on the
weird.
right.
On the right side,
Window,
yep, I see that.
window.
Thank you. We'll have a quick look at that as we go by. Right there. Still got lots of these little, what we're considering to
Yeah.
be forams, these white dots that you see coating the bottom, just incredible densities. Let's be sure this isn't a coralliid. I think it is a primnoid.
It's a primnoid.
Yep.
Another little one. Very
Yep.
tiny one. Yeah.
There we go, for sure.
Oh, yeah.
Yeah, so I guess the polyps are more dense on this compared to the one we collected, and this is much more planar.
Oh, yeah, absolutely.
Yeah.
Right. Yeah. That one that we collected, I don't think we've seen before.
I wonder if that's a yellow bryozoan down to its left.
Yeah, yeah, I know. Talk
These
about
bryozoa-
small.
Yeah, these bryozoans are so small,
Yeah.
they're really hard to see. If they don't come up on a rock, you're
Yeah.
not going to get them.
You can do a white balance.
If you think that's necessary. No, I'm satisfied. Okay. There's yeah, so that's a hydrocoral all the way down in the lower left corner. Yep.
Okay.
Kind of looks like the top.
It looks like the
So
top,
flat,
that's for
but
sure.
We still
Yeah.
got 40, 50,
40,
60 meters at
59.
least.
So
Okay.
it's definitely.
Oh, so this isn't the steep part. We're still in the not very steep part.
There's a plateau. This
Yeah.
is just a plateau, a shoulder, as it were.
It's the Grenadier, yep. Grenadier up on the right. There's one of these anemones. We can get a side view of it as we pass it. This is the one we saw earlier. It had the sides sort of curled.
Oh yeah,
Yep.
big brown.
Yep.
What's that noise back there? Yeah.
We can keep going.
Oh, somebody's
it's like a
using a
drill.
wrench or something. Yeah. Power screwdriver.
Oh, yeah, we may have passed a cookie star. We did see one earlier in the dive, and then maybe up here on the left side we're passing one. This is a left
What's this?
Royala, I think.
Oh yeah, no, left way. Oh yeah,
Yep.
right. Yep. Right. Correct.
Yep. Urchin on the side of the rock on the right. It's like a dead sponge skeleton. That thing right in the center. It's kind of greenish brown. You know, one of the things we're doing is we're really focused on the sessile communities. That is, those animals that are attached to the bottom, grow up off the bottom, and are taking advantage of the food that's moving through the water to some extent. And so because of that, it's really important that they're found in an area where the currents are sufficient to sustain them. And that seems to be making a difference. So on YouTube, Lost Hero asked if we saw a cookie star. I think I'm seeing several of them now. What's this thing over here, Gerard? Top left, little ball. This looks like a barrel sponge, maybe.
Yeah.
Is that like a geodid or something?
Yeah, it's kind of what it's looking like from this distance.
Huh. We haven't seen one of those before.
No, exactly.
Looks like Acanthascus.
Say again, Sarah?
Acanthascus?
Acanthascus.
Sponge.
Oh, a rosellid,
It's
okay.
a glass sponge? Yes.
So a glass sponge roselidae.
That claterize it is.
This looks like more of a glass sponge, don't you think?
That's what she said, yeah. Glass
Yeah,
sponge in the
nice.
family roselidae.
Oh, a rosellid.
Yeah,
Oh,
so I was
yeah.
wrong about
It's
the
weird
geodid.
because it has its
I was
sections
yeah.
inside out.
I see.
You can see the spicules create kind of a weird netting around its out exterior.
So glass sponge hexactinellida, you see a white primnoid coral to its lower left, and there's a hydrocoral in front of it tucked in the rock, and then a brachiopod, that sort of bivalve thing off to the left.
And if you go a little bit left, there's a fish in the hole.
Oh yeah, I see it, okay. A codling or something.
Hmm.
First I'm going to look at the sponge.
Yeah.
Get a good view of that.
Oh, that's
You can
nice.
see what Sarah means about the spicules. The 6 radiate spicules are kind of inside out, so it looks like there's all these needles on the outside that kind of trap things in there.
Well, and I know from personal experience that they go into your hand quite
Yeah.
well as well.
So some worm tubes and things on there. There's some amphipods. Alright, so now we'll look over this fish that's tucked down in one of these little depressions. There he is.
Oh, look at there. Wow. Ah, that's
Is
amazing.
that a codling, you think? Focus, man, focus.
I don't know. That's. You see if you can get a closer shot of that thing.
Its tail looks like an ophidian tail.
It's kind of grumpy.
Very grumpy. But nestled totally
Yeah.
into the rock. That's amazing. What's
That fin looks pretty. It looks almost sculpin-like, the fin.
Yeah, we need a laser. Where are the lasers?
I don't think so.
That fish, like, had that little tiny talent when I spun around.
Yeah, that's not what it looks like to me, but that doesn't necessarily mean anything. I'm not a fish guy.
So do we have a size on that?
I'll pull out and look at the lasers. Are the lasers on? Okay.
The lasers in there somewhere. Oh, there they are. Wow, that's
very small.
5 or 6 centimeters. That thing's not very big. Wow, huh?
What is the purple thing?
Look at that.
Up to the right, there's some
Looks like it's damp.
That's
Purple thing
amazing.
Swimming
to the right.
up to the right.
Squiggle.
Huh.
Up a little.
Up, the other up. Yep, sorry. Yeah. That's a polychaete, I believe.
Squiggle seems to be.
That's a what?
That's a little worm, scale worm.
Oh yeah, that's the scale worm.
Quite impressive.
Yeah.
Ah, it's colorful.
Oh, that's very nice. Yeah. Hmm. Hmm.
It's way out there.
Oh, there's big
Yeah,
fish.
no, it's far. It's pretty. The deep sea may be dark, but you bring lights down here, and it's really wonderful, the sort of spectrum of color that you get to see. All right, moving on.
Yeah, Amy mentioned that there were a lot of Swiftia in the Gulf of Alaska seamounts, and I think we saw plenty of Swiftia up in the Aleutians also, and that was one reason why we thought we would see plenty of Swiftia here.
Agreed.
But
Yep.
that's the first one that
Yep.
we've seen so far.
Well, but we've also said this repeatedly that we know that a lot of this fauna has a patchy distribution.
Right.
And one of the things that's really tricky as a scientist when you're coming out here with this very expensive equipment in limited time is what place do you pick, and did you pick the right spot to drop down on? Because
Right.
maybe just over the next ridge
Yeah,
is all that stuff we're looking for.
That's right.
So,
that's right.
Yeah, you get a little bit lucky.
There's a fair amount of area at this depth on this seamount, and we're just taking a look at a very tiny amount of it.
Yep.
Yeah, that's right. So the things we're missing right now, like the Paragorgia that we haven't seen yet, could be here, but we just may not see them on this dive.
I guess, you can imagine you were going hiking in a national park and you were told the entire national park you could go the equivalent of 4 football field lengths. And that's it. That's all you got to see.
Yeah, that's
so
right.
of course you're going to miss a lot, and that's equivalent to what we're doing out here. Let's look at this yellow
Yeah.
thing.
You might not see the canyon in the Grand Canyon National Park.
Exactly. Right. You missed the canyon because you were off looking at the rim or
Right.
something. Yeah. That's a really good
Yeah.
analogy.
And Scott, unless I just want to let you know, I did a quick test. We're getting about a 35-second latency or delay from what you say that when it comes up on YouTube.
Okay, thank you.
Okay, good. Yeah.
Is our audio at least correlated to the imagery?
Yeah,
or is
no, it's matched up. Perfect.
Okay, all right, great. So if I ask them to answer a question, I have to wait at least a minute.
Yeah.
Yeah, okay. So this is a cluster of zoanthids, this colonial anemone, and these are the ones less referred to before as incorporating foreign material into their body to make them much tougher. And earlier we saw one overgrowing a primnoid colony, and I wonder if that's what the fate here was. There was a primnoid under there.
Oh,
Now
yeah.
it's just zoanthid.
There was a primnoid under there. Yeah, right,
This
exactly.
almost looks like the Paragorgia.
Yeah.
So that's a no.
It looks like that style, but we saw this on a primnoid earlier, and there was a little bit of primnoid branch left.
Yeah.
And it was all these gold specks Tina
Look at these.
suggested were the
These are,
sclerites of the primnoid in the zoanthid.
Okay, zoom in on that amphipod on the left-hand side if you
Right.
can, if you can get any more zoom.
Several of them.
This is a really interesting amphipod. This is a eucyrid, and the reason I can tell you that is because in the eucyrids they have the anterior appendages and the mouth parts are purple. They're always purple, and I've seen them purple everywhere from the northern end of the world to the southern. And it's really interesting to actually see a couple of them right here on the zoantha. They're very distinct.
There you go.
Yep.
That looks like Henricia on the left.
Henricia, the genus name for that sea star that has those long arms and not much of a body.
I gotta put that in here. Henricia. I think we saw it yesterday too. If we did.
Which one? Henricia?
Henricia. Yeah.
Naples and Bristol.
We don't think the holes that are in the rock that we're seeing here are produced by the worms. They're pretty old erosional features, I think. Maybe that were formed by rocks moving around there or some such thing. Our depth hasn't changed very much over the last
No, it hasn't
quite
changed
some
much
time.
at all in quite a while.
Yep. On a flat part of this slope, it did go up a little bit and then it went in, and now I think we're going to go on a fairly steep
That would
wall.
be good if we did that.
Yep. We're trying to get there.
Yeah. A lot of cobble in this area.
Yeah. Well, maybe that's good. It means we're at the base of a wall, maybe.
Yeah, exactly.
Great, you have a new waypoint.
Cover that, man. Thanks.
We're a few meters up above the bottom here, so you can get a good broad view of all this rubble. Yep.
More fish.
Look out the distance.
They're going to slide up starboard quite a bit here, a little bit closer to this. A little strung out, so slide close.
Okay.
So we're going essentially due north, and does the current look like it's coming from right to left again?
No, I'm just lateraling pretty hard till I start.
Oh, I see. Okay.
I'll be able to get you starting here a little bit.
Are the squares in the screen meaningful to the waypoints? Meaning, can I interpret the distance between the waypoint 2 and 3 by knowing the size of the
They're
square?
100 meters.
100 meters. Okay, so we're a couple hundred, 150 or so meters from waypoint 3. Okay. So we're about 150 meters or so from the top of that ridge.
Oh, okay. Good.
You gonna look at whatever that white thing is?
Yeah, I think that's a sponge, so we can go check that out though. Verify. It's a Walteria for sure. So this is a glass sponge. You can see it's branched. Then it has these sort of spines arising from the main part of the body.
Funny branch tips. Yeah, lots of branches.
Yep.
Many branches.
Some very small, maybe Anthomastus recruits down here in the front center.
Baby Anthomastus.
Yep. And then there's a snail at the base of this Walteria sponge. Some worms living on the inside of that sponge.
It's already got the worms in it.
Yep. You can see the sort of whitish brown. You can see them translucently. The sponge branches make these sort of hollow tubes that the worms live in. Looks like there's also a brittle star maybe living on the other side of that. And then there's that snail.
It's like a Calliostomid snail.
I'll take your word for that.
I'm only going on the basis of how shiny it is, but it might be something else entirely.
What's is this I guess those are amphipods, and then there's an anemone in the background. Yep.
Yeah.
Okay.
That's that spiny amphipod again.
Yep. Another small Anthomastus colony.
Good to go?
Yep. Thank you. Lafroyella sponge. Some urchin. A little less rubble on the right side. Another grenadier ahead, another branching Walteria sponge. Oh, there's, first Paragorgia coming up. Bubblegum coral coming up. There it is.
There it is. We must be near the top.
Yep.
Paragorgia.
So this is what we saw yesterday from the same depth. We started seeing these very large Paragorgia colonies, which is interesting. We didn't see a lot of small ones. We saw a few, but
yeah,
every once in a while we come across 3 or 4 of these massive colonies.
Well, maybe we'll see some young recruits
the
up
lasers
here.
on right now. Oh yeah, there they are. Whew. That's a big base. Those 2 green dots are 10 centimeters.
Yeah, maybe we'll see some young recruits further along here.
You can see some brittle stars in the branches, and then you'll also see some purple strips. Those are polychaete worms. We saw that yesterday as well, and we collected a smaller Paragorgia, and there were several of these worms on it. So it looks like these worms like to live associated with these Paragorgia. I can see at least 8 or 9 of them. You think you can try to get the lasers towards the base so we can see the diameter of this thing? There we go. So that's 6 or so centimeters diameter, maybe.
How much?
Maybe 6.
6.
Yeah, you see the lasers there right now?
Oh, there they are. Yeah. Oh, wow, that's big.
Yeah, that's very big.
Wow, that's like 20 centimeters across.
The holdfast, but
the holdfast
right,
is,
the actual diameter of the stalk is probably about 6 or 7.
The main axis, yeah, like 6 or 7. Yeah, right. That's quite large.
Zoom in and have a look at some of these polychaetes.
You want to sit down and have a look?
I kind of want to keep looking, not staying, because we've seen this so much. But
yeah,
you notice all the polyps are retracted in here.
Yeah, must have heard us coming. What is the depth range of the OMZ at these seamounts? Yeah, that's a good question. I don't think there's a strong OMZ here at all, actually. I think it's a continuous decrease in oxygen down to about 1.5 or something, but I'll have to go back and check on that.
These worms are scale worms, family Polynoidae. So not a huge amount of diversity of
No.
associates. Basically, there's two species. There's one kind of brittle star, and then there's one kind of
Yeah.
worm. Nothing else.
But what's interesting is that this Paragorgia arborea always has these same species on it.
Yep.
Yeah. And I think that the worm on here actually, I think, is probably something new, because when I pulled the one off yesterday from the colony, it was extremely flat. I was very
worm?
surprised. Yeah, the
Yeah.
worm was just so it's made a little groove on the colony for it to sit in, but the worm itself is very, very flat. It's like maybe a couple of millimeters thick, and that's it. I mean, I was really surprised. I've never seen a scale worm with that kind of thinness to it.
Another thing worth noting here is that you see all that rubble in the background, but this colony is growing on a much more stable surface. And this colony is probably, I'm going to guess, a couple hundred years old easily. And
Yeah.
so it has to be growing on something that's stable, or else it wouldn't be able to hold it in place in order to get this large.
Yeah, that's right, exactly.
Quite beautiful. Quite lonely. It's very interesting.
And
it's the only
lonely,
colony
yeah.
out here, you know, and
Yeah,
who's it going to mate with?
but there may be more off in the distance here,
Oh, yeah.
along the top of this ridge or
No
whatever.
doubt, no doubt.
Yeah. But
But if this was a great place to feed, you'd expect there'd be a whole line of them.
yeah,
You know?
that's right. Yeah, exactly.
So the question is why? Is it just because the luck of the draw, or
Yeah.
you know, maybe when this thing first settled, the currents were optimal for it to grow when it was young,
Yeah.
and now it's big enough that it can survive. But maybe there's not a strong enough current for other young ones to survive.
Yeah, that could well be.
Can we next check out this thing right there, that white? It's probably one of those sponges we saw earlier.
Yeah, I think that's right. The rubble is probably moving. I mean, that's why you don't get stuff on it, especially anything that lives for a really long time. I think that's basically probably the issue.
So this is another one of these stoloniferous sponges. What's
Yeah, exactly. I don't know what that is, but
that bizarre? Yeah, that's exactly what it is. So I guess that might be a Paragorgia recruit.
yeah, that's a
Yeah.
that is. It's
Yeah.
a Paragorgia
There you go.
recruit. Yeah,
That's a good
and
place
there's
to
a
settle.
solid on the wall there.
Yeah. Two of them.
Two solids up, another one up top. Yeah.
All right. So first, I'm just going to look at this.
Yeah,
sponge
that's
just
a baby
quickly.
Paragorgia right there. Look at that.
So that sponge in the background we saw earlier, and every time we see it, it seems to grow like this. You know, sort of the old man's beard.
Yeah, exactly.
But
mine,
this is
brown
really
on the top
interesting.
and white on the bottom.
So we were just noting that very large bubblegum coral and saying, why aren't there a whole bunch of little babies? Oh, and there's another Nudibranch.
Yep.
A different Nudibranch.
Yeah, a very different
on the right
nudibranch.
side there with the orange
Why is the
serratia.
Why does a nudibranch at this depth have colored
Go figure.
cirri or whatever they are?
All right, one story at a time. So the bubblegum coral, this is a very young recruit. You can see where the polyps emerge.
The other
Wait,
thing
is that a bubblegum coral?
yeah, that's definitely a
Okay.
bubblegum
All
coral.
right, we're all in agreement there.
Yeah.
So
I
that's
know
again
you're
one.
probably thinking it could have been an anthomastus.
Right, yeah. It's just, there's
Because of the color and whatever, the
Well,
polyps all being in.
all the polyps
So-
in plus the
Yeah.
siphonozoids.
Yeah. And what is that? A brittle star with its legs sticking out
think
up
so.
underneath that rock?
Yep. It's exactly what that is. But
It's
this,
like
Paperweighty.
It's like they're hermit crabs of brittle stars. I'm gonna wear a rock to protect myself.
what's happened to my focus here? There we go.
I think we should get
that
a
nudibranch is
a
amazing.
really nice close up on the
that's as close as I can
nudibranch
get.
if we can.
Bigger
That's-
one
that's it.
above the Paragorgia.
Yeah.
on the left.
Okay, thanks. No, that's a solid sea cucumber.
Yeah.
No.
More left?
No. So straight up from the Paragorgia. You can just barely see people.
Oh, sorry. Yeah. There you go. Thank you. That's a nice one.
Ah, there's a bigger one.
Yeah.
Oh, good eyes.
There we go.
Yeah.
Yeah, so those orange extensions that you see are
Wow.
the orange filaments are literally extensions from the gut, and they end at these little white tips, and those tips are cnidosacs, where it's storing the stinging cells from the cnidaria that is eaten to use in its own defense. And its head is kind of pointed away and to the left. You can see the two long sensory tentacles. Very cool. So that's a cnidarian specialist, cnidarian feeding specialist. And then up on the rock there on the left, you can see two of them. These are the solid sea cucumbers that are specialized to live on the walls. Great. Should we try to suction the?
Huh?
Should we try to suction the nudibranch?
Are we what?
Should we try to suction the nudibranch?
Ooh, they're too hard to preserve.
Okay.
They're very
All right.
hard to preserve.
We can
actually.
move on. Thank you.
Yeah. Interesting idea, but yeah. John, could you put that profile back up with the oxygen in it? Oxygen profile. That was interesting looking. I didn't quite get a chance to catch it, but yeah, there you go.
There's some question about why the rocks have different color. John, are you able to say anything about that?
You know, I just came in a little bit ago. I was looking at this. These look like you know, the similar type of sedimentary or volcaniclastic stuff we saw earlier, only it's not really exposed well. But when we were doing the close-ups of the creatures, you could kind of see that. And then all these, the sediment, the sand is different than we've seen before. I wonder if we could suction some of that. We'll take.
Sure. I mean, we want to get a sediment sample from Flex. Anyway, here she is. Yeah. So, are you okay to get some of this? You want some of this rubbly stuff?
Yeah, this brownish stuff. Yes. I mean, we can always get some other stuff later. We got 8 jars, right? So.
Yep. So, how about a sediment sample? And we'll take the rubbly stuff not in one, so one of the others, and then we'll do another one later.
So this is not one. This is not a fine method. Correct. So I can go to.
Yeah, we'll reserve that for later when we see a finer
I haven't
grain
seen
sediment.
much of the finer sediment, but
Yeah, this is more. Yeah, put this wherever. It's just
70 microbals at 2,000. Yeah, that's the about 26.
Yeah, these are those kind of flat, platy, slabby rocks covered with the crust. So that's it's different than the lava rock. But when you put the crust on it, it's hard to tell the difference sometimes.
No. Right.
It's good. Sorry.
Yeah, any of that stuff.
Coming on with it, right?
May take a little more to get it. Ooh.
oh, it's
Oh.
finer underneath.
Oh,
There
look
you
at that.
You don't want the white stuff, right?
Well, no, we want both. I mean, John wants.
Yeah, it's made.
the-
Didn't expect it. Didn't expect that.
And I think if you're where you're going into two, most of that white stuff isn't going to stay anyway. So.
Yeah, the fine mesh isn't on.
Yep.
Catch it.
Okay. What did I miss? Anything? No animals while I was over there.
No.
Okay.
This is YOM 1061 slurp tube.
Interesting how the sand gets a bit aggregated in there.
Interesting.
He's cementing the grains together. Yeah, I mean, so I asked John to bring up that profile on a higher resolution. I think that the issue is that there is a minimum there, but it's much above the summit of the seamount, so the seamounts aren't experiencing low oxygen values at all. I think that they're in pretty well oxygenated water.
It's already emptied out of there. I'll have a look at jar tubes. Sometimes it's not either.
Oh. Dirt.
How much do you want?
Which one is it in? 8?
2. It's in 2.
Can you use this stuff then?
Yeah,
So,
I thought it was all just rubble, but
Yeah.
finer stuff underneath would be really good to have.
Do you want us to
Yeah,
go
so
to
get-
one as well?
I think so, since we're here.
Okay.
I like that.
yeah.
So is that good for 2, or keep going for 2 for a little bit and then go to 1?
Yeah, get a little bit more and then call it. I think we just need a little bit of it, see what it is. Get another blast of it.
So the comment from Lost Hero, yeah, I think that's the thinking, that the white sediments that you're seeing here are probably pelagic derived. That is, they are the shells of pelagic organisms that have died and sunk and accumulated on the seamount, where the larger dark rubble that you're seeing on the surface is erosional from the seamount. Is that
Yeah,
correct, John?
it's interesting that it's obviously the the dark stuff is more recent and later, but it's kind of it's interesting that it's not like layered in
Yeah.
between. So it's definitely kind of happened in one episode, not alternating.
And I don't know if that also means that something about the current sweeping through here and the way it's
Yeah.
kind of winnowing the lighter sediments off the top.
Yeah, I mean, could have had a big slide event or something that shoved it all down, but
Good to go to 1 now?
Yes.
yeah, that's good. Thanks.
She's looking for the lighter stuff.
Calling this 106-2 back.
Okay. So where was the OMZ?
800 to 600 meters.
Oh.
I was tuned out of that part of the conversation. If you're chatting with Amy, I noticed her comment about the siphonozoids, and I did say something. Trying to see it belching into the canister there.
Yeah.
Yeah,
Where?
it's fine.
You think that's good? Or you
Well, I
want-
certainly saw a lot belching down in there. I don't know if it's good the way it's going there, but I'm certainly pleased to see it's going into the canister. It's coming in pulses.
I think one more nice, good scoop should be enough. This looks a lot coarser, or at least some of it does, than what I've been dealing with, so.
Alright, so is that gonna be enough? And you're gonna move from here?
Yes, except that there was one thing I wanted to look at before you left.
Okay. So then just hold on to that.
Yeah.
Go ahead and look around.
Oops, I'm sorry.
Fly out. Power.
All right, it's a little hydrocoral there, I believe. Yep. I don't know if that's Crypthelia, but it almost looks like they're little Venus flytraps. Each of those little containers has several hydroid polyps in it. So if you've heard of fire coral, for example, this is related to fire coral. Excellent. Did you get a frame grab of that?
Yep.
Thank you. Okay, I'm good. I'm going to leave the camera for you while you get stowed. All right, so we took a couple of sediment samples there from 1,458 meters depth here on Yomie Seamount. We've been spending the day climbing from kind of the floor of a plateau and up onto a ridge that's on the top of the north end of Yomie. And we're still kind of heading up and wondering if once we get to the top of this ridge we'll see more corals. And we just saw our first large coral of the day, which was Paragorgia bubblegum coral. And yesterday in similar conditions when we got to the top of the feature we saw lots more of those. And so maybe that's what we're in for.
One other thing we want to do, Scott, we want to, it looks like 8, might got popped open.
Okay.
So we want to secure 8 again.
Copy. I'm just going to do a little bit of maintenance on our quivers, make sure we don't lose any samples that we've already caught up with.
There was a clog. Is there anything in quiver 9?
Not yet, no.
Well, our plan was just to push this one in deeper than we normally would.
Okay.
So it'll.
I didn't know if you guys wanted to like stow that elsewhere and then just use 9 to clog 8, but whatever works best for you guys.
I think getting it pushed in.
I think that
Yeah, just not so deep to break the sponge that's in there, but
That should hold right there.
I think that ribbon sponge might be a sponge
okay.
called Ocelastra.
We write it down for Leslie's keeping notes.
POE CIL LA STR.
Okay. I'm going to call Old Man Beard Sponge.
It doesn't quite look like it has an extra fringe to it that's weird, but Ocelastra is the only thing I can think that grows in ribbons like that.
Yeah, it's kind of interesting. I remember seeing much larger ones in Central Pacific.
Another bubble gum coral coming out.
Why am I not seeing it?
I don't think, is it?
That's the one we just left.
Yeah, okay, yeah, that was it.
That landscape looks like images from the 1970s on the surface of Venus.
Bridge Avenue
Of
Waypoint.
Venus.
Oh, yeah.
Thank you. Very nice to meet you. Thanks for the northbound 37.
That's what Sarah suggests is that sponge. That's kind of like the ribbony sponge.
Oh, really?
It's about, they're saying about 0.9 foot.
There's a whole bunch of this fringy hydroid growing on the rock again, like we saw somewhat deeper. Lots more of these, what we're calling forams, these little white dots, calcareous forams. I guess I should look that up and make sure that there is such a thing.
Yeah. You can always drift backwards if you're struggling to get to us. Okay.
You think all this rubble has come off of this little platform that we're seeing?
I don't know. I'm looking at it. Not necessarily.
Okay.
It's an interesting outcrop.
Yeah.
What is this first
It's like
letter?
it's mantling this
P.
ridge.
P. Silastra.
Piscilastra?
Astra.
Astra?
Yeah.
And that's a sponge?
You criticizing my penmanship? Yes, that was a sponge.
Some people
Yes, that is something, that's a bubblegum coral. Just
Some
in the
people's
distance,
art-
it doesn't look the
Some
right
people's
color.
artwork is better than their penmanship.
Yeah, thanks. All I heard there was praise for my artwork.
Yeah. Sponge, huh?
Yeah. That, I don't know, let's check that out. I'm thinking it's a very large branching Walteria sponge with crinoids growing on it and maybe some hydrozoans growing on it. Alright, not Walteria, it's that other one. The uretid with the tubes and the zoanthids, right? Yeah.
Keep going.
Yeah, we can keep going.
What is it?
Uretid sponge? Uretidae?
I cannot hear you at all.
Grenadier. Maybe on the far left, I see a sponge in the corner there. Let's just fly by that, see if it's anything different.
So that was a uretid?
Yep, there's
The
a
last
baby, another baby Paragorgia bubblegum colony
Oh,
on
yeah.
the right side.
So there are some recruits here.
Yep, there's another one. It was a sponge and I got it, so I'm okay with that. And there's another baby Paragorgia there, so we got some recruits. Left rail of sponges showing up with more frequency. What's this yellow thing here?
What is that?
It's a hydrozoan.
Oh yeah.
Yep. That's a dead sponge, dead sponge, dead sponge. So those brownish green things, these are all sponge skeletons. The tissue has died and so they're no longer white in color. This is a baby Paragorgia down in there. Bunch of recruits. There's an
A lot of rubble. Cobble, boulder rubble here. Oh, that's a mastis. No? Yeah? No? Wow.
I'd say that's an Anthomastus.
There are several little Anthomastus colonies.
All right, that's good.
They all landed in the same place.
Yep. Travel in packs. Maybe a small primnode colony tucked in there. Here's another small
Boy, this is all
baby Paragorgia
Yeah, here we are. Yeah.
on the left. Grenadier, sponges, sponges, sponges. Is this the same Lefraella?
It's funny how we went from that
Yes.
sort of cracked up pavement type looking stuff to this.
Yeah.
Yeah.
Let's have a look at this one, J-Rod. And whoever's flying.
But we saw that brown coarse sediment between.
That guy right there in the middle?
Yep.
That coarse brown sediment is also still around in between the rocks.
Yeah.
As well as some lighter stuff.
That might. Well, it's definitely primnode. I'm not sure if
Bunch
it's
of
like the
dead
one we
sponges.
collected or the one we collected yesterday.
What are all the little dots?
We think they're foraminiferans, or I think they're foraminiferans.
Should we, if this is the one we haven't collected from the seamount yet, should we collect it?
We can certainly pick up that rock. I guess it's an easy one to collect, huh?
Hey, there you go.
What do you think, J-Rod?
What about that collection?
Yeah, you think we can just pick up that rock with it and put the whole rock in a bio box? Let me get some good views of this first.
Barnacles.
I might have to move that.
Okay. So this one would also have anemones. We would get one of these brachiopods. And I'm not sure what all else is just under right here. Maybe even a solid. It's not too big, right?
We'll give it a go.
Thanks.
All right, good for me to set up.
Yeah, turning over to you. I did do that. So while we're collecting, I see a question on the YouTube chat. Do onshore scientists watch the stream like they do on Okeanos dives? And certainly we have alerted our colleagues ashore that we're doing this work, and some of them are tuning in. And so there is one scientist right now on shore who's also following on YouTube and sending us some comments and requests and questions, and we're going back and forth with her. So absolutely, it's one of the valuable things about this model of doing science at sea and being able to stream the video live to shore, is that we can get interaction with our colleagues and very quick feedback. That may have been that Pseudomonida.
Yeah, that's what lobster just took off.
Yeah, but it might have been the Pseudomonida. It was too. It might have been the Pseudomonida. It was too colored.
Ah, yeah. That's true. We'll never get it.
Possibly.
A nice place to hide down in there.
Moving towards it, we'll let you know, depending on the sample.
Okay, thanks. I'll just bring the heat around a little bit to slow down the sideways movement. It's easier to stop.
Yeah, it just keeps going, doesn't it? One of those benthic jellies there. Is that benzicodon, I think?
That sounds right.
You want to make a note of that in the annotation? You notice that little jelly that's swimming around there? I think that might be benzicodon. It spends more time settled on the bottom than it does swimming. Oh boy, talk about a tenuous grasp.
That jelly's been determined to be a part of the story.
Well, it's too bad that bryozoan wasn't on this rock.
Yeah. That's a pretty good sized rock.
Yes.
So, are you thinking box 2?
Yeah. Yes, I am thinking that.
Start pointing out then as you come up, alright? I'm just slowly pointing out. We're just going to this box right here. So just keep coming up nice and easy.
Scott, is this main sample a rock or is the main sample is the main sample rock or is it wild?
I would just leave it. It's going to a big file box. I'm in upper. And don't drop it in. Set it in. So once you get in.
I think it's going to set on the corals.
But it also means the coral's in the box, so that's fine.
I can punch out a little bit more if you want me to.
Don't expect these big rocks all the time. Nikki, you remember when I said that this would be an easy way to collect it?
2-gallon rock. Bigger bags.
I shall call this iceberg rock.
You're doing great.
It's kind of a nice effect having that red light on.
Let it go.
Let it go.
So this is 107-1.
Nice. Still
right.
standing. Nicely done, man.
That's good.
That was a little more challenging than I thought. Sorry about that. But everybody's breathing again. Nice to see. One of the exciting things about that rock is that there were a number of different things on it, including the coral, the associates that were on the coral, but there was also a brachiopod, which we've been seeing very commonly, and that was on the rock also.
Yeah.
So that'll be a
to
really
have
good
the-
sample.
Have the brachyrod.
Got just to confirm.
Yeah.
That was Blair 107-1
Correct.
from Noah plus associate rock.
Correct. In BioBox 2. And maybe if you have the space, you can also put brachiopod, BRACHIOPODA. Thank you.
Good to keep moving.
Yes.
You can ask Marcia if she's still looking at that. She is. You can go ahead and move the waypoints to the sheet.
J-Rod, have you overridden my iris control?
I did.
All right. That's okay. All right. That's good.
There's just fun or something hanging out up there.
Let's check it out. If that's a sponge, it's a big
Something
one.
big.
Yeah. I think it is one of those. I keep forgetting the tube name.
Wow, look at that.
Oh, Euretidae. Yeah, thank you. Good thing you.
Oh,
Have a look at the staff wall.
Just a quick look. We've seen several examples, but this is a nice large one.
oh, that thing is gigantic.
So I guess. It's like a Galatheid crab, squat lobster tucked in there, lots of zoanthids. Yep. Not much room left at the end on this sponge.
No vacancy.
No vacancy. Some brittle stars. Okay. You can compare that with the top right. There's one of the Lefroyella sponges that has the anemones in it. Okay, that's good. Alright, what am I missing here? Level 5. No, dead sponge, or live sponges. This one, we could get one more look at this. We've only seen one of these today. This barrel-shaped rosellid sponge. This is another kind of glass sponge. Pretty different morphology from the others. Just get a quick look. That is indeed what it is. Amphipod, less
Oh yeah, there you go.
So swinging
Yeah,
just
let's see
to
that.
about, what is
Yeah.
that, 7 o'clock from the sponge. Down below, there's this little lip you see sort of wiggling back and forth, and there are 2 amphipods on it. And these are amphipods they build, these little mud sticks. Let's see if I can get that in focus.
It's a pair, but no babies, huh?
No babies that I can see.
Yeah.
There you go. Okay, that's good. Good go, J-Rod. Let's see. Yep, ready to go. Large anthomastus, I think, in the upper left. One of those zoanthid covered sponges on the right. Walterius sponge on the left. I think there's a hydrocoral right here in the just to the right of center.
That probably weighed me down a little bit.
Start moving. That's
1,432 meters depth.
So there's a sponge off the line. Sure.
Hey, Murray, so you have a new waypoint?
Those big sun stars in the center right.
Roger that.
It's another spongy dive, Les.
Yeah. This is sponge country.
There's a black coral in the center.
Oh yeah.
This one's different. That's different. This is a new one. Let's slow down and have a good look at this. This might be a Bathypathes. Or was it Dendropathes? No, not Dendropathes.
On the right, it looks like the.
On the right, I think it's Dendrobathypathes.
Yeah.
But on the left,
There's another black coral over there.
yeah, I think that's
Yeah.
the Dendrobathypathes.
Oh, that's the dendrobath.
that,
Yeah. Right.
forms that sort of wall.
This one's got branches coming out in all lots of directions.
Right, and what I'm trying to see is it's
But-
really hard to tell. Is there a central axis? And
Yeah.
these are all just coming out in like pinnules, but it's twisted?
Yeah.
Or are there multiple axes
Yeah.
that they're coming out in?
Can't even see. Sort of can't even see the hold fast.
No. It's like going down and away from us.
I don't know.
It's a pretty good image. There's certainly no smaller secondary pinnules in there, and these pinnules aren't branching, so that's going to tell us something. Solid sea cucumbers again. All right, I think that's okay. I'm still looking to get up to the top
Yeah.
before we do.
How far up more? Oh, I see where we are. Yeah.
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 Let's see if I can find that in my database. There's something like that coral that we just saw called Parantipathes, very different from the more bottle-breast Parantipathes that I'm used to, but that was from the Aleutians, so could possibly be that. That's a small black coral, the Dendropathes. We've seen that one in here. There's a pink sea star. That was interesting. Yellow feather star.
Out of the rubble and onto the smooth.
Is that a resingid? Let's look at this one. This is the first one we've seen on the right side here, that star-like thing. That's a resingid asteroid.
It's not a Brazingid, it must be a sun star.
I don't think so. I think it's a resingid. You can see the little side pinnules on the arms.
Oh yeah. Oh, look at that.
That's what that is.
Huh. A lot of busted up arms.
Yep.
A lot of regenerating was.
Well,
a lot of regenerating
right,
arms.
they've been nipped off and they're growing back for sure. This is a sea star that's kind of adapted to feed in the water column by raising its arms up, capturing food particles with little hooks on those spines that are coming off the arms. Thank you.
Every now and then they get nipped off.
Yep. Fish come along and take a little bite of those arms and they can grow them back.
Pretty massive sponge.
There's a sun star.
Yeah, there's the sun star. Yeah.
There's another one of those anemones with the brown tentacles. The large euretid
Well, there's several.
sponge. Yep, that's a black coral.
Huh.
We can move on from that. What's this one? Yeah, what's this white thing? Is that like a primnellid? I want to look at this one more closely, please.
I think we should take that rock too. Is that primnellid?
Don't joke like that when John's around.
What is that? That is a primnellid. Right?
No, I think that's a Walteria with really long spines. That's what that is.
The Korea below it.
Yep. So.
Oh,
yeah,
that's weird.
So that's a Walteria sponge that has a hydroid growing off it in the upper rock, and then this is a Furiidae sponge. This sort of
a Furiid.
what's the word I'm looking for? Frilly.
Yeah, the Furiid.
And then you've got the one that's kind of the tubes where the laser is right now. That's the Euretidae. So those are 3 different kinds of glass sponges right there. And actually, I think that the brown sponge that you see may be dead
Yeah, we
Furiidae.
collected that one yesterday.
Yeah. And then what's this red thing back here? Is this a baby Paragorgia? I guess it is. Just starting to put out some polyps there. There you go.
Oh, look at there. A youngster.
A youngster. This
Another
is
youngster.
Amy before was noting the darker dots that you see on there. Those are the siphonozoids, the ones that sort of channel the water into the colony.
Not too many polyps on that one.
Behind the Furiid sponge, where? Oh yeah. So on the right side there.
Mm-hmm.
And there's several of them actually. Just new recruits. So this is an example of larva has settled and started to grow, and these colonies have to start somewhere. And they start with 1 polyp. So you can see the topmost one looks like there's 1 polyp, and then just below it there are 2, and then in front of that there's 1. And then I see 1, 2, 3, 4, 5, 6, 7, 8, 9 more. So it looks like some recent recruits. Yeah, that's not a tube anemone. That's a burrowing anemone.
What about the brown one?
Yep. Edwardsiidae.
What's that?
The brown one, Edwardsiidae, I think is the family of the burrowing anemones.
Ah, okay.
And then I believe that's a bryozoan, the white branching structure up on the top. And then again, these little white dots on the side of the rock, I'm calling them forams, but I'm not 100% convinced yet that they're not just tiny hydrocorals, so I'm going to have to get with Steve Cairns on that.
I mean, are we looking to come to a stop on this waypoint?
No.
Probably not.
Yeah, we're good to go now. There's one of the hollow tubes we finally found out was a Serianthid tube earlier today.
Oh, yeah. That's almost in a place where you could get it.
There's another one over there. I'd like to know there's somebody in it.
Yeah. Yeah. Well, they could be in there,
Yep.
just pulled in a long way, which is
Sure.
Syrienses like to do. There's
Sure.
another bigger Paragorgia.
Doesn't look like there's much in the way of associates on this one. It's got some worms.
But it was interesting yesterday that we saw those one of those tubes with a scale worm in it, though.
Yeah.
Yeah.
Which would suggest there wasn't an anemone.
Right. Yeah. Exactly. Yeah.
There's another bubblegum coral coming up in the distance.
Oh yeah. So now we're getting into the right neighborhood.
Is this a smaller one down here, J-Rod?
Mm-hmm. Mm-hmm. Hmm.
Something else.
What is that?
Maybe a black coral, maybe a basket star. Anybody want to make wagers before I zoom in? Okay, the black corals have it.
It's like black coral, huh?
If you put your money on black coral, go collect your winnings.
I put my money on black corals.
Huh. What a mess of branches on that thing.
Yeah.
Jeez.
Well, the messy black coral,
Yeah.
that one.
Yeah, that's crazy.
Huh.
Not 100% certain I know exactly what that is. It's not exactly in a wall format, you know?
Yeah.
It's like it doesn't know how it wants to branch.
Actually, this one is confused. That Bathypathes like thing looks confused.
Yeah, yeah. This is the confused slope. I guess we got the top of a sponge over there. Okay. Moving on up.
Onward.
It's going to be disappointing if we get up there and there's nothing up there, and we ought to come back down here and get them all.
Oh, we'll go down the other side.
Well, yeah.
Where's the lanterns?
Well, we passed the 1,400 meter mark. We're now at 1,395 meters. Yep. It's kind of a scrawny colony, isn't it? Not a lot of branches.
Yeah, probably still young. Hasn't filled out yet. Doesn't have the middle age spread going.
Yeah. Let me just zoom in on this. These lower branches, it look like there's some white tips. I wonder if something's been feeding on it, or looks like there's a bit of damage. See what I mean, Les? It looks like this branch has been broken off, maybe. I don't know, or if it's newly growing. And down here, it looks like
it's got scars.
Yeah, that's what it looks like. Or maybe that's just a polychaete on the lower side. I don't know.
That's a worm.
It's a worm or an anemone?
It's a ring anemone.
Yeah.
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 There might be another one starting to grow up above there. What's that? Oh, it's just a mucus streak.
Alright, my autofocus is playing games again here. J-Rod, I think I've lost the focus again. I can't seem to, it doesn't seem to matter which way I turn it.
More amphipods on the whip.
See if I can do it.
Okay.
It definitely likes the background.
You see this?
Yeah,
Is that
I
what's
was
producing
noticing that. I was
the
thinking,
web?
when we were talking about vermidage yesterday. Looks more like a vermidid net.
Okay, so you got that on the manual now?
Yeah, it's on manual. You should be able to just turn your manual without.
No, I don't think so.
It's not focusing.
It's not, no, well, maybe. It's now I'm, yeah, okay. I'm changing the focus to back there, so it worked. It worked. I don't think that's a vermitid because they wouldn't have tentacles coming out of the hole like that. So that's going to be a worm.
Yeah, right.
That's more like the terabellid or.
Yeah.
All right. So I wonder if they're the ones that create this sort of spider web stuff.
Yeah.
We've been wondering that for a long time.
Yeah, it's a real nice net.
Yeah.
But I don't know who's making it.
Well, maybe they're making it.
Huh?
Maybe they're making it. Those worms.
Yeah. Maybe.
All right, we're good to go.
Thanks, man.
Thank you.
See you there, really excited.
Little black coral. Another black coral. You know what? Is it too late to go back just like a meter? And look on the left side, please.
Left side.
Yep. Lower left corner. There was a reddish orange colony, and I said black coral, but I thought I saw something in its branches. Yeah, so it's just come into view right there. I just want to be certain that I'm not overlooking a Paragorgia. I'm not. Okay. So it's a black coral, but it seems to have a...
Always black corals.
Yeah, but it looks like it has an ophiuroid in the branch, and that's not normal.
Yeah.
This is not normal.
You want to get in close for a tight zoom?
No. That's okay. Thank you.
Big coral coming out. Weird, they're all by themselves everywhere.
Yeah.
Not like yesterday when they were pumped up.
Black coral, Paragorgia coral, another black coral. That's more like the wall that we saw and collected yesterday, the Dendrobathypathes. There's another one up there. Mastis. Yes, please. So what have we got so far? We've got bamboo whip, a stoloniferous octocoral, 2 primnoids. That's it. Yeah, we can check that out. Looks like a very small black coral. They look so different when they don't have a lot of branches compared to when they do. Yeah, this is before it's gone all crazy. No question, that's what it is. So that looks like the Dendrobathopathes again. There's another one. Walterius sponge, the white branching structure on the left. Several Lephroiellas here on the right. And I'm not sure. Let's look at this one here, the bigger one. This might, I'm not sure. Is this new? Yeah. Is that also a Russellid?
No, that might be Corbitellini,
Corbitellini.
I think. Possibly.
Cookie cutter star and a cup coral up there. Yeah, let's do that. Thanks.
A solid to the left, and another one up there. There's more solids. There's another one. Three solids.
Any particular view that I need, Sarah?
Any particular view?
Yeah, I mean, is this good or is
That
it?
is good. If you can zoom in the edges,
Okay.
kind of in the upper right corner,
Okay.
that might get a better idea of the structure of the spicules.
So, solitary hydroid on there.
I don't know if you have time, but I recommend taking a piece of this.
Okay. All right, we're gonna do a collection here. Well, we just want to kind of break a piece off. So, a piece that you get your claw on and you're sure there's something on it. That's all that's really necessary. It's going to be sort of crispy.
Yeah.
And I think that it would be relatively easy to dislodge the whole thing. So, however you think you can get the claw on and then just snap a piece. You can do it.
We're going to crush it.
Yeah, it's going to be fairly brittle.
Yeah, it crushes, but I think you can crush enough just to sort of break some. So long as you can't squeeze it the whole way down. And we can put this in a quad box.
Quad.
With sponges like this, we have had success breaking pieces and slurping it,
Oh, okay.
like if it's that brittle.
Yeah.
So if you can't get a claw on it, that's an option.
Okay.
Chris says it's not Corbitellini, so something else. He thinks it might be also a different kind of uretid sponge.
Okay.
It's a hard sponge.
But he's still interested?
Yeah.
Okay.
Oh,
There. Oh, yeah. That's nice. That's
I don't
perfect.
see the piece.
Perfect. Excellent. So I'm going to put Euretidae as the sample. Okay. That's EURETIDAE. Biobox 1B is full, but otherwise we got 3 spots to go.
Anything other than B? Correct?
Correct. While we're sampling here, I'll remind everybody who's watching, this is a feed from the Schmidt Ocean Institute's ROV Falcor. We're on the Emperor Seamount's expedition looking at coral communities and where they transition from a northern fauna to a southern fauna. Oh, I see there's some polychaete worms in with that sponge. That's nice. We are currently diving on Yomie Seamount. This is the first ever dive, ROV dive on the Seamount. It's in Biobox 1A.
108-1?
That is correct, 108-1. We're good to go after this. Yeah, so we're on Yomie Seamount, which is towards the north end of the Emperor Seamount. We're at a latitude of 42 degrees 26 minutes north, 170 degrees 26 minutes east. You can see the inset here in your screen is showing you a view, a close-up of where we're diving, and now we're pulling out, and you can see the seamount, which is an underwater volcano in a long chain of seamounts that runs from north to south, going from about the Aleutian Islands in the north and going down towards the northwest Hawaiian Ridge in the south, that then extends towards the southeast towards Hawaii. And we're interested in the oceanography that are associated with the change in the communities and the water masses, and so we're sampling coral communities, microbial communities, lots of water samples, and be doing the analysis on this to see where the change occurs and why. Let's have a look at this crab on this black coral here. This is kind of interesting.
Ooh, tall stalks.
Yeah, wow, that's a big Paragorgia coming up, but
Two anthemestes.
I want to see the crab that's on this black coral, and once again, I can't seem to control the iris.
That's because I took all control and I was the last sample.
Nice. Total control.
Just go to camera, take off auto.
Yeah, look at that.
So this could be a chirostylid, but I was just informed there's a newly
Thank you.
described family in the Chirostyloidea. Let me pull it up.
Yep, that's good. Thanks.
So this is what we would have called Gastroptychus iaspis CF, but it could also be sternostylid, sternostylus.
So I'm also noticing now there's definitely 2 black corals we're seeing. These ones have really small polyps, and the other ones have much larger polyps. And I'm thinking, I guess when we collected that wall of black coral, it must have been like this. So the other one must be different. How much of a face view do I need for ID? I guess the spines that are on the...
Spines are always useful.
That's a nice dance.
Alright, nice view.
Does this have a bare stalk like an umbellapathes? It's hard to tell.
I'm
from
not
this
sure.
angle.
Yeah, it's hard to tell from this angle. I don't think so. All right, I'm going to look up the samples that we've already collected, but we can move on while I do that. Check out this giant up here.
You ready to go see the giant?
Yes.
Oh, that's a tall Paragorgia, though. Oh. Look at that. Hmm. Wow. Look at the size of this one, Scott. Oh, we need to get a look at the crab on that one too.
Okay.
On the back side of that. Yeah.
Want to look at the crab?
Yeah.
Yeah.
And so I don't have the black corals that we collected yesterday to be able to compare to. This looks like the same one that we've been getting an image of that's been walking around.
I think so, but I think we need to get a good look. Oh, it's a lithodid. Yeah. What's it doing way up there? So it also has regenerated a couple of legs. What? There's some fish predation going on here.
Yeah, there's the lasers. So 10, 20, 30, 40. Wow. That's probably like that branch alone was probably about 30 centimeters to 40
Across
centimeters
there?
just across there.
Yeah. So you think this colony is what, about a meter and a half high?
Yeah.
I can probably tell you if you want.
Yeah, let's zoom out here.
It is big.
Come up and tell you about how tall it is?
Sure.
It's about my height, I think.
Can I take the camera for a second?
All right.
Look at
Camera's
that.
yours.
Huh. That's amazing. I wonder if we're getting into an area where they're dense enough to do water bottles.
We're almost at the top. We might be at the top of the ridge, actually.
Almost.
We're almost at the top of the ridge.
Oh, okay.
A couple meters.
Okay. Maybe that's why we're picking up more of these, huh?
Yep.
So,
How tall do you think it is?
I think it is 1.5 meters tall.
yeah, that's kind of my guess.
Oh, look at that. 3 meters?
About 3 meters?
What?
I'm a little bit higher than it. No, not.
2.5 to 3 meters high.
It's how much?
2.5 to 3 meters tall.
Whoa, really?
That's a big sucker.
That's rather large.
That's
2.5 to 3
extending up
meters?
there. Yep.
Huh. That's amazing.
Yep.
It is big.
Pretty magnificent.
Yeah. Beauty. So the base of this thing must be almost 10 centimeters at the base, right?
Yeah. Can I take the camera back?
Maybe it's right closer so you can see the way it's going to face you.
There we go.
Oh yeah, look at that. A bit more. Yeah. Huh. Yeah.
Yeah, that's big. Lasers are 10 centimeters apart, so that's easy. And that's as a side view. I'll bet it's even wider from the other view.
Yeah. Good thing there's not much current. Plus, the brittle star that's on it is different than what we've seen.
That's
See
the
that
one
brown
that I
one?
expect to see. That's the one that we see in the Atlantic.
Yeah, that's an astroschematic.
Right, exactly.
Yeah,
That's
exactly.
not what we have been seeing.
Right. Huh. Wow. That's very interesting too.
Yep.
They're the more typical ones for here.
Yeah, associated with Paragorgia for
Yeah.
sure.
If I had an astro schematic out here.
It's almost like I got a line of them. So there's one in the distance.
Yep.
Huh.
And there was also one when I was facing. Yeah.
Yep. We can move on when you're finished with the big shots.
Go ahead and go uphill.
Yep.
Do you think it's dense enough to do an eDNA signature?
What's that?
Do you think it's dense enough to do an eDNA signature?
We're almost at the top of this ridge
Okay.
where we expect them to be even more.
Okay.
Huh. Oh, gee. That's nice to see those big old ones every once in a while.
Yep.
There's some young ones.
Yeah, we're
Some
starting to get
youngins,
into
yeah.
higher density here now.
Teenagers, if you can call an 80-year-old coral teenager.
Well, compared to
Absolutely. I agree.
another
It's all
big one.
relative.
Another big one. Literally, right?
Literally.
Comes another one.
I'm sorry, can you just back down a little bit? I'm looking on the lower left just out of the screen. I want to be sure that I'm not missing a Bathypathes. The thing that the fish is flying over?
Side view of it.
Thank you. Yep. Nope. There we go.
Huh.
Let me get settled down real quick.
Looky there.
Thank you. Thought that looked a little
nice.
different. So this may have been, you know, we saw that one that was all kind of twisted up on itself. It may have been a much bigger one of these. It was all just kind of twisted. So I think this might be worth sampling. We don't have anything like this yet.
Okay.
You good for a
I'm up
sample?
for that.
Are we in a position we can do that?
There's the other kind of black coral just to the right.
Yeah, I saw that one. So yeah, this is the one that has much bigger polyps, longer tentacles per polyp.
Yeah.
Looks like our quiver mount might have broke.
The quiver what?
Mount might have broke.
Oh, the mount. Oh, is it hanging down?
Looks like it's swung.
Yeah, yeah.
Let's see what the head. Just
Yeah.
just over there. Looks like I was maybe a hose thing.
That's the profile from the top.
Those are both sealed. They're behind. They're still good. Looks like the top one broken. That's usually a repair. All right, so we're taking a sample of this, right?
Yes, please. So essentially, this has one upright stem with those side branches, so we just want to like cut maybe the top.
Where are you going to want it to go?
Quiver?
The other way, it's no good.
Quiver's on this side?
Either side.
Okay, just, yeah, if you feel comfortable moving that in and out, yes, quiver is good. Very interesting. But you said you weren't really interested in black corals?
Just because we weren't going to be doing a lot of rigorous sampling of them.
Yeah. Yeah, I think it's going to go in a quiver easily.
Okay. I guess I'll figure out. See what we got there. Set up should be good for these. Those bolts came out. They were tapped. So I ripped them out. So we just put all the bolts in there. About 10 years ago, we used to-
So we collected 2 black corals yesterday, right?
Yeah, we did. Yep.
So one was that.
Huh?
One was the Dendrobathopathes. What was the other one?
I thought the one was from the wall.
Yeah, one was the wall.
Yeah.
But did we get
The
2?
other one was a very open branch thing.
Yeah.
We'll just hold those until recovery.
What's happened to our scribbles?
Yeah, it's like,
Come on over.
so only 8 and 12 have something in them right
Just
now,
those
right?
two.
The only 2 that have samples of.
Yeah, they-
Oh, I see. They're the 2 front ones. Yeah, I keep forgetting the orientation.
What are those still hanging on by?
Both mine.
You didn't want to hear thread, right? Call this Bathypathes. Phypathes. I don't know enough about Bathypathes distribution to know if there's different species north and central Pacific. Or I don't know that they know well
Yeah,
enough.
I don't know. Yeah.
Oh, sorry, I haven't been paying attention to all the questions that have been coming in on YouTube. So you asked about the yellow at the bottom. Yes, those are feather stars, crinoids. There's a joke about the Latin names that we're using, and I agree they can be confusing, but they're really necessary. And there's some pretty good examples of why that's the case. The common names change from place to place and language to language, but the Latin names don't. It's an agreed upon system by scientists all over the world, and they're used in publications and descriptions. So when we use the Latin name, we all know exactly what we're talking about. The common names, we don't. So that's why we will break into Latin, and as much as possible, we'll then try to translate it into a common English name for you.
Scott, the top is okay?
Yep, so you see that there's a central branch going down, a central axis.
Yeah.
If you count down, let's say 6 or 7 of the side branches, just going in there and clipping the top. But you've got to clip through that central axis because you need those side branches. This will give a lot. It's very floppy. It's fine. You can sort of whack it around, but ultimately you will need to cut that central branch.
Too much sitting. Can't do so much.
think I see some something like corallimorphs down there. I lost where we were looking. Oh, I was talking to Sarah. Thanks.
Does it look like it has sub-branching at the base? Or is it just a bunch of tentacles?
They have polyps that come off the central axis that are longer.
Yeah.
And I think that might be what it is. As you probably know, often where the polychaete hangs out.
Yeah, those long tentacles are confusing.
It does kind of look like that's a branch, though, doesn't it?
Yeah.
Maybe there's another small colony down there. Yep, that's plenty. More than enough. You might want to just wait a second because I'm not sure that you've got the center in the cutter. Yeah, there. Now, yeah, it keeps like blowing out on you. That's good. Yeah, that looks good. Yep, yep. Did it?
Not all of it.
No, I think you did it. I think you cut off the top. That's perfect. Yeah. So there's still a discussion about the Latin names. If it was named in English, there would be no confusion. That's only true if you speak English.
What about Old English?
Oldie English. I guess you have to go to the person who designed the system, Linnaeus. He was Swedish, but chose to do everything in Latin, and so that's the standard we still follow. But it is true, you could have used any code in any language, but once there's an agreed upon system, you got to stick to it.
Once we let well, myself and a Jed as well, and I'd argue that his name is spelled incorrectly in English.
I will say at least that we don't speak Latin to each other in our labs. We only use it for the names, really. I think you've got it protected enough that it's going to sink if you open it slowly. Of course, what I think and what it wants to do are two totally different things.
I need help.
What's that creature that's weaving with that branch? Did you see that? There was something wrapped around there. Oh, it's a floater.
Oh.
It's a floater. We should open the bio box.
That gets anywhere near my solution sample. I'm trying to figure out.
Or the bio box. Oh, it's gone.
Might get it. Just might get it. You okay for those receptors?
Yep, absolutely.
Now we're away.
Yep, it's right there. It's right below you. Now it's lodged, I think. Is it on?
It's right below your section.
What do we have? Section 8.
It's right underneath the suction gun.
Yep.
Just come back up a little bit more. It's not moving now. Okay.
That's getting it stuck.
No, you're good.
Very nice. In 8. Let's watch it come in there.
Turn my suction down to 1.
There it is. All right.
Suction's off. Index.
That was 109-1 is in section 8. Yeah, let's get to the top of this. Nice work, Ben. I'm sorry, it's a floater. I should have known better. And there goes the quiver.
Actually, I had it, but
Do we have iris control again?
CTD.
Or do
As
I?
soon as they leave your body, I'll pull these guys.
There you go. What's that guy right there?
I think that's the wall of black coral. The one on the left? Yeah.
Keep going up.
Yep. It's very impressive.
Okay.
There's another Bathypathes like what we just collected. And the sponges, which it's a good sign to be seeing some different stuff here as we approach the top of this ridge. Anthomastus. Bridge control.
That time's still 1800.
I believe so.
I think that's fair. That would be leaving at about 5.
Yep. Yes, that's correct. 1800. All right, thank you.
Just gonna center you up a little bit. That's okay.
Yeah.
there's a.
Yeah, I'm looking at this little pink thing right in the center here for a second. It's okay. It's a very small bubblegum coral. That's fine.
I got a
Okay.
forest coming up.
All right. Oh yeah. Several large bamboo coral. Bamboo. That's wishful thinking, huh? Bubblegum corals. Large
A little bit of a
ones.
Freudian slip.
A little bit. There's another young recruit here on the right of center. And here comes the massive.
Oh, there's a group.
Yep.
Oh, look, urchins too.
Yep. It's looking familiar, huh?
Does this count as a mini garden?
No. But I know it's not.
Yeah.
When you don't have an actual one, you just eat little pots of it.
A copse. I believe a copse is the right word. That is a skeleton. Yeah. Hey, oh no, Brendan, you don't use the Paragorgia, huh? No. Yeah, that's a skeleton of a Paragorgia. You see, the tissue has been lost and it's all white on the inside.
Scott,
Yep.
see, there's one back there as well.
Yeah, you can see it. Yeah,
Here,
yep,
yeah.
yep. And there's one further back there.
You can tilt down a little bit. I don't want to fly too low with those clippers.
Well, maybe we should do bottles here.
I think we should do it
Yeah,
just
that's a good
one.
idea. That's a good idea. So maybe we
least
could
one.
settle and get a Niskin sample.
Just on this side of it.
I don't know that it matters if we think the current's coming across like this. That'd be fine.
Do you agree?
Yeah, the current's definitely changed up here. Significantly.
Okay, bottles.
Yep, we're doing it.
All right.
Start off on the left side.
If you get a chance, I think
5
there's
colonies.
a sea star that just went off screen left.
Yeah.
It looks different to me.
Okay.
It's left of
On the bottom.
Yeah, on the bottom.
What's that?
A sea star on a rock left of the corals.
Sarah sees a different looking sea star on the bottom.
Ah,
It's like the third one I've seen. I just want to confirm it.
we know
That's
which
what
way
it
the
is.
current's running here?
Coming from the north.
Yeah, I think it's
Coming
coming
this way?
across.
Oh, it's from the north.
Yeah.
Oh, nice. Oh, nice.
Yeah, so somebody's asking, there's a lot of rubble and it doesn't look stable, but where those giant corals are must be stable or they wouldn't be there.
Yeah, that's right, exactly. And not too much current either. You can't get a 2 meter or 3 meter high coral towering up like that with one current.
I'll be right back.
Okay.
Nature break.
Exactly.
So 110.1 is this Niskin sample.
110.1 is what?
Yep, this water sample.
Okay. I suppose he means by that I should write it down. Okay.
You know which bottle is fired.
Oh, 110.1. Is that
Last
what
time
you
I
got?
tried to trigger 4 and it works. Don't write it down
John
yet.
110.1. eDNA water sample by Paragorgia.
And then
That's okay. Oh yeah, I need the water bottle numbers. That's right. Okay, so 0 is bottle. What bottle number is that?
no, it's not. 3.
Is that 4? Niskin 4?
No.
No.
1. I think it's 1. You want 4?
No, I don't care necessarily. It's just right next to 3 that I don't know in order. Or I can't see the sticker numbers.
No, I think you're right. It looks like there's a 2 right next to there.
Okay.
Yeah, yellow should be. Here,
You ready?
Okay.
I'm pulling you around.
Yeah, you're fine. There you go. All right. We'll let it go. All right. We put it in that. That was number one.
Number one.
Yep.
Another one?
And the next one will be 10 meters up
10 meters above this spot?
off the bottom.
Okay.
About
So we're at
flat falling.
40.
There we are. Oh, it's over there. If you can grab it. 42, 26. Whoops.
Flatfish down there. Saw flatfish.
I've been filling that out.
Huh?
I haven't been filling out the lat longs.
Oh, okay.
I get it from the event log.
I had instructions.
Oh.
All right.
I was just like, fill out all the things.
Good work.
So,
Get in there, Jay. You can't get out.
Yeah.
Got to tilt to it.
store on the sample.
It's just the 10 meter above bottom one now?
Yes.
This is 10 meters above bottom. I see what I did here. I wrote 10 out one.
They get different sample numbers though.
Oh, okay. Yeah, you erase all that other crap I did.
Pulling.
Fire bottle. Fire kick go. Alright, going back down?
Which one was this?
That was
2?
number 2. Going back down or?
Do you also want the 20 meter? Or?
Yes.
Okay. You want the 20 meter also?
Yeah.
Going up.
Standing by.
Was the first one in 1?
Yep. 1, 2, 3. In that
Thanks.
order.
Okay.
Alright. Ready? Yep. Okay, fire and deploy.
Alrighty.
Gonna go back down?
Yep.
That's good. What time do we are off bottom?
About an hour.
5 o'clock. Well, we're just about ready to turn and go up the ridge.
Mm-hmm.
Now we're in the Paragorgia zone.
I'm curious about just looking over the other side,
Yeah.
or at least on the upstream side. All right, so we just took some water samples in an area where we saw several large Paragorgia bubblegum corals. So we took a water sample just downstream, and then we went 10 meters above the bottom, took another water sample, and then 20 meters above the bottom, took another water sample. And each of those samples will be examined for DNA in the water to see if we're able to detect the corals just by their DNA that's been sloughed off into the water column. And if that is successful, it means that in the future, from ships, we could deploy water sampling devices without having an ROV and learn something about the corals that are living in that area. So that's the general idea behind that science. Okay, so where was over here, Sarah?
There's actually one just off screen if you tilt down.
Okay, to what, right or left? Or?
Down, tilt down.
I want to look at a sea star that's...
To the bottom corner there.
Oh, that one with no arms?
Yeah.
Okay. You see that,
It
Gerard?
kind of looks like it has webbed arms.
Yep.
In the family Asterinidae.
Okay.
I think, but this one might be different. He's kind of fat.
Looks kind of long in one dimension. Weird.
Yeah, no, it's more likely something like Hyposteria.
Little stoloniferous coral next to it.
What do you think this is?
I don't know quite, to be honest.
That's why you wanted to have a look?
Yeah.
I'm wrong, that's not a stoloniferous, there's a bunch of little hydrocorals there.
Interesting.
I think
It's got a lot of papillae sticking up on this sea star.
it's good imagery. That'll be good to send to Chris Ma.
Okay,
Thank you.
you're welcome.
Yeah.
Yeah.
Try to sit down and come in.
I think,
So it's definitely not a goniasterid.
yeah,
Could be an
I'm okay.
Asterinidae, but I'm not sure. The only ones we have records of are super shallow.
See that big bite out there?
Yeah, it looks like we were seeing that yesterday, the A cesta. So there's one tucked in there on the left side, tucked way in there. Interesting. There's another baby Paragorgia, young recruit, several solid holothurians.
You have to move on?
Yeah. So some similarities to what we saw on Suico, this depth with the Paragorgia at the top and the urchins. The urchins aren't as abundant as they were, but they're here.
Would you go down there and get a Kit Kat?
Yeah.
Oh my God. Lethal.
My first one of the trip.
If I have a Kit Kat piece, I'm gonna want to go down and get a whole Kit Kat.
You won't, because I took the last one.
Huh?
You won't, because I took the last one.
Ah, all right then. You saved me. Yesterday, I think you got the last fig bar too.
Clearly need to go down before me.
Look at the size of these.
Yep, it's impressive.
Wow, that's really impressive. Paragorgia forest. They're right up on this ridge, aren't they? They're just lining the ridge, actually. What are we facing? 309. Yeah.
Huh. That's quite a line.
Yeah. They're just lined
So,
right up.
do you know, are you, let's see, so
Yeah.
the ROV right now is sort of pointed in the orientation of this axis. So we've been climbing this ridge from basically towards the north, and now we have gotten close to the top of it, and we've turned somewhat looking along the ridge crest.
The current's coming from my right to the left.
Okay, so it's sweeping over this.
The current's coming over
axis,
the
yeah.
ridge. Yeah.
So these bubblegum corals are kind of near the top of this ridge crest. Oh wow!
Oh.
Oh,
Ice cream. Oh,
there's a special delivery here. Good stuff. Thank you. Yeah. Well, this is really cool, this linear arrangement. And you can notice the other thing about, I'm going to lighten this up, so you see several of these bubblegum corals going along and up this ridge crest, but they're all, the wide axis of the fans are all pointing in the same direction. So they're all intercepting the dominant current that is coming from the right across to the left. There's no accident that that's where they're doing it best. There's another group of them just behind and a little bit lower on the crest. So Les, why are the urchins congregated below these Paragorgia? Are they feeding on something that's coming from the Paragorgia? Is it possible that the Paragorgia are interrupting the flow? Where?
Talking about the fish?
Mucus.
Oh yeah, yep, I see it.
Oh, look at that. Oh wow, a rockfish. Oh darn.
I'm gonna zoom out so you can see better. Done here.
You want to get in closer to it?
Yeah, if we
Yeah,
could
get a
just
little
get a
closer
good
view.
view of the head there.
It doesn't want us to, but we do need to.
Just got another
God, look how thick that base is on that.
Oh yeah, they are really amazing, aren't they? There's a lot of little ones over in the back row.
This might be okay.
Yeah, it's a rockfish. I don't know which species lives at this kind of depth, but
All right, that's good. There's one of those corallimorph-like or anemone-like things in the rock. Oh, that was convenient. Thank you. And then there was one more thing as we were zooming in. What's? Oh, it's just a hole in the rock with a brittle star. Okay, let's stop fighting that. Thank you. Yeah, so you're seeing the currents are basically pushing the ROV from right to left. It's telling you there's a pretty good current in this area, which is allowing these Paragorgia to thrive and get so big.
part.
Sure. Where those urchins are, you mean?
Yeah, like at the base of it.
All the white, you mean? I think that's sponge. It's that uretid sponge, I believe. We can go towards that big Paragorgia with all the urchins at the base. There's one knocked down. Uh-oh. The urchins are after that one. Look at that.
Yeah.
Urchins are all over the skeleton
Oh,
of that
yeah.
Paragorgia. So you can see where the
Oh,
tissue
wow,
is
look
still
at that.
alive, where there's red tissue, and then where it's white. I suspect those urchins, and maybe that sea star, have been munching away on it. There's even a bunch of snails down there.
There's a star there too.
So
Cleaning it right
this
off.
is certainly providing a good meal for a variety of these benthic organisms.
That's a grim picture.
Yep. Grim
See
if
the
you're
sea
the
star
coral.
underneath?
I do see the sea star. I mentioned it when we
Okay.
were coming through.
Yeah. There's a
Couple
sea
of cup
star
corals
under the
down there
edge
too.
but
Yeah.
I can't tell.
Everybody's seeing the sea star.
Yeah.
Getting a look at this snail that's on the underside of this branch here.
Oh yeah. There's a cup coral.
I'm just trying to
Okay.
touch down.
Sure. I will wait for you to touch down before I do it anymore.
Yeah.
Doing those crabs, Les.
Oh yeah. Huh, young crab.
I almost thought I saw eyes on this snail. For all the YouTube fans of Chris Ma, our operation is such that we don't have a phone, so Chris can't call in and give us information. He can email or post to the social media, but we don't have the ability to take the phone calls, so that's why you're not hearing from him.
Well, we were saying actually, wow, for both the coral and the ice cream.
Yes, we were. You notice it got really quiet in here for a little bit.
Yeah. We had an ice cream delivery from the galley, which was quite happily received.
All right, so once again, there's many things feeding on this, and I'm taking this opportunity to look at this snail because it's got an interesting angle on it. There we go. I see it's not the eye, I guess it was the siphon that I was seeing just next to that red tentacle.
It's gonna melt in there. Container.
Yeah, no, I got it in the bag.
Oh, you done?
Yeah. And let's look at one of these urchins.
So I don't know what snail this is. That's the thing.
Yeah, there's several of them.
I'm inclined to think of it as a caliostomid in terms of the family, based on the shininess of the shell and the tightness of the body whorl and the. But I don't know, and it looks like actually the one in the upper part you might be able to see the operculum.
Yeah, I think that is what
on
we see.
that, and that's can be a helpful character as well. Oh yeah.
you
So
can
when
also
when the snail pulls into the shell, then the little door that covers it over.
You can also see the sort of almost spongy texture to this colony where the red tissue has been removed. You see some of the holes, and basically the live tissue would be continuous throughout the kind of, I described it as concrete, the way the skeleton is sort of fused together, sclerites, but it remains, it allows for these tunnels to pass through, so all the polyps can be interconnected to one another. You can see the tube feet of these sea urchins kind of waving around. They use the tube feet to grasp onto food, transfer it to the mouth, and for moving themselves over the substrate.
Well, I guess the other big question is what happened to topple this
Sure.
Paragorgia colony?
All right, so why did this get knocked over in the first place?
Yeah.
So I think we answered our own question about why the urchins are hanging around below the Paragorgia. They're waiting for it to drop branches.
Yeah, maybe they drop things. But
Yeah. It's kind of like waiting for your favorite restaurant to open, but you may have to wait about 20 years.
They can't climb up.
Yeah, we've seen urchins on vertical corals, but I haven't seen any of these on the urchin, on the coral, so I guess not.
well, and the other thing is we did see a Paragorgia being fed on by this starfish Evoplasoma, and in the Atlantic, where a similar situation where the Paragorgia had been knocked over somehow, and the sea stars were all over it. And then 18 months later, when we went back, the sea stars were still all over it. It was taking a long time,
Very long
for them to eat the tissue, and
Yeah,
they
so that-
actually hadn't progressed very much, like maybe a centimeter or two along
Exactly.
the colony. I mean, it was
Exactly. So what we were
amazingly
just
slow.
seeing just now, that may be 2-3 years of feeding.
Yeah, or
and
more.
may continue for
Yeah,
another
right,
few years.
exactly.
There's a lot of food there.
Yeah, because things do happen more slowly at this temperature.
Yep.
But the urchin that we collected one of those urchins yesterday, and that urchin was still alive in the bucket when it came up, but it wasn't moving very rapidly. That may be its normal. I don't go very fast. And I think we also should point out that the spines on those urchins are extremely sharp, very
Yeah.
fine tip, and they penetrate things quite readily. Now, who do we have here?
Is this a coralliid here on the right of center? Ruh-roh. Ruh-roh. Stand by to collect.
So we have a lithoid crab.
Yeah. If it'll fit, yeah, sure. Let's get a closer look and see
look,
what
the
we can
crab
do
is
about
having
that.
something to eat. See what it's. Okay. I'm gonna see what the crab was deciding it was going to chew on. So guesses on the coral? I don't know what's a coral. But
I think it's a coralliid. It's a coralliid or a different kind of Paragorgia.
I
yeah,
think it's Swiftia, Scott.
Really?
Mm-hmm.
Oh, all right.
It's moving.
it's a swifty.
Well, that is true.
It's a what?
That is a good clue. They think it's Swiftia, and they point out that it's moving, so it's probably not a coralliid.
Yeah.
This crab just can eat it before we can look at it.
Well, I was heading for Swiftia, but because the Swiftia pacifica has got a very different morphology than the
Yeah.
rest of the.
Right. They have
species in the
this
genus.
appears to have relatively thicker branches than I'm used to seeing on the Atlantic Swiftia.
That's right. Yeah, they're much more dense.
Yeah.
Yeah. We have to wait for the crab to go by, I guess. Don't ruin our spec. Get your leg off of there. First, you have to get walked on by a crab, and
You will never let you forget who you use.
yeah, if that's a Swiftie, I guess we better take it because
Yep.
that's the northern. That's another northern indicator.
So what we were just discussing is whether we want to clip a piece to go into a quiver or if we want to pick up the rock.
Oh,
So let's check out what's going on.
oh yeah,
with the
that's
rock.
definitely a Swiftia. What else is on there?
Yep.
Baby fish.
Oh, fish.
Amphipods all over the.
Sorry. Yeah,
What is
it's a
that? Look at
little
them all.
Christmas tree of amphipods. And there's
That's
a
on the
caprellid
Swiftia, dead
amphipod as well.
Swiftia. Huh.
Yeah, so that's the Swiftia skeleton that you're seeing there that's brown.
Well, look at all the amphipods on the live part as well. See all the little lit up eyes? The lights are coming off all the eyes like little lights.
And there's also the one with the spines down here on the right.
Huh.
And the little anemone, ring anemone.
So it actually would be pretty good to get the whole rock if unless the rock is too big.
It's also got a
but-
stoloniferous octocoral on the rock.
Yeah.
How do you feel about the rock size?
It looks pretty big, but I mean, we might be able to.
I think that's bigger than the last one.
Well, if it's bigger than the last one, yeah.
I think it might. Well, I think more of it's exposed this time. It's probably the same size as that.
Okay. I think there's enough amphipods and stuff on there that if we were
Yeah, well, I
cut at
think
the base,
I
we could
agree.
get
I
it
think
all.
that's probably it.
So let's do a clipping and go into a quad
They
box.
may half of them may swim away
And
anyway.
I may ask you to see if you can grab that rock because it's got that solid.
I just cut it as close to the base as possible.
You can push on that or wait for you to get down.
Meanwhile, the crab is continuing his wanderings.
Yeah. Well, I'm glad we found the Swiftia again. Earlier I was talking about the difference between an axial skeleton and a consolidated or concrete-like skeleton. So this has an axial skeleton. You can see that brown part of the skeleton that's exposed is that axial skeleton. It's protein-based, so it's really quite flexible. And then the red tissue is the living part that's covering it. So in this case, the skeleton is not living, and the continuous tubes of the live tissue do not extend through that skeleton, unlike the Paragorgia bubblegum corals we were seeing.
Do you want me to note this one as Swiftia? Or
Yes, Swiftia, yep.
Go for a deeper bite.
Yeah, I think if you push down a little bit further. Yeah, that looks good, Peter.
That should do it, yeah. Exactly. Good.
That'd be one way to collect the rock, huh? Come on, you've got to be kidding me. The power of the holdfast.
Oh, that's amazing.
I mean, if you think that whole thing can drop in, right? Okay, all right. That is, a new way to do geology.
You know what they always told you when you're collecting in the intertidal zone? It's always turn your rock back over.
1C and D are empty and all of 3.
Scott.
Yes.
We could collect a smaller rock over here.
Okay.
If there's another one, or whoever, from the top of this ridge.
Try it. I'd fit.
They're like
If it
Christmas
looks like
tree
it's
decorations.
4 is empty, and that's fine. Not close to the end. Okay.
B1
A and B
B
are
has
taking
got
C
something
and
in
D,
it.
so after open. How come I can't see the sponge?
I think it's just one that was the wall.
It was just a piece, wasn't it?
All those amphipods are completely oblivious to what is going on. They're all just hanging right on there, like,
I don't know, this current got picked up with something, I don't know what it is, but it's amazing. They don't take off.
There it goes, dropping in. BioBox 1D as in dog.
YOM 111-1.
Correct. Copy that. Yeah. Thank you.
Generally, we see that is that the things that live on the corals like that, who have some kind of strong association, don't tend to take off when the coral gets collected. They come all the way to the surface with the coral.
John?
Yeah.
Do you like this rock? Because I really like this rock.
What's growing on it?
Cucumber. That one right there.
Oh gosh.
It's kind of big.
Oh, really? Is it?
Scott, you're looking like a geologist more and
John,
more.
Sorry. No, it's only
you
like
can't
12
say no.
Okay. Is that? You think that?
Get it if you want to get it.
I do kind of want to get it because I've seen quite a few of these.
Yeah, it would be nice to get that.
See,
sold.
J-Rod says it's not too big.
All right.
Yeah, that could go in BioBox 4.
You'll have to go in about which is open for.
4. Yeah.
Yeah.
I see it. Yeah, right under 31.
Oh, I see.
You name it for?
I'll try.
This, yeah. Yeah, that's good.
Oh, nice. One solid coming home. Watch the soul. Look at the soul it's pulling in. It knows something is up.
4. BioBox 4.
Oh, that's what it was. See that?
Yeah.
We saw something on a rock that was brown like that and circular like that.
Yeah, we weren't sure what.
But we didn't know what it was. Now we know it. So it was a solid that had pulled the tentacles in. Huh. Very nice.
4. Five, even, I don't know. Let's give out. I suppose if you think of alternatives.
Someone
Solid is P-S-O-L-I.
Oh, yeah, but they're not that standard. They're standard. So, going in 4, or do you want to try both?
If you think it'll drop easily in 5, then go for it. Won't move around much, that's for sure.
I'll open them up, both up for you. Thank you.
That's 111-2, if that wasn't obvious.
That's the middle.
Yeah, in response to the comment about depth perception, I think that's absolutely right. But one thing that you're not seeing that the pilot sees is there are views from other cameras. And so they can use that, the pilot then can use that to judge better where to put the manipulator. And in this case now it's putting a rock in the box. And there's a camera that's not online to you that where it's looking straight down, so you can see where the rock is being aimed. So that's it is on the Facebook. Is that on the Facebook?
It's on the YouTube. Yeah.
Oh, that's on the YouTube. Okay, yeah. So now you can see how the pilot can see different angles, get an idea of where to put things.
Nice collections there. So that's good for your rock up here?
Yeah.
Yeah.
Right. Cool. Continue. We got another 25 minutes or so.
That's good.
Would you mind imaging the sea star on the bottom left?
Okay. That one that's coiled over the rock?
Yeah.
All right.
Just to get it.
We just want to get it.
Just enough to see the texture on his arms.
Sure.
It's hard because he's so light and the dark rocks are so dark.
Get a good look at this cup coral as well. Another solid next to it.
Oh, there's another solid.
Yep.
I know. Is that a 4-armed star or has it got 5 arms?
5. There's one
one's
going
tucked
back
in the back
towards
there.
the bryozoan.
Yeah, tucked in at a weird angle. Yeah.
All right. So,
Huh.
English names: sea cucumber, flat sea cucumber on the rock. A sea star, folded up its arms around the rock. A cup coral in the front, and then a bryozoan, which has no common name,
Yeah.
back there.
Want to be cool.
Thank you.
Yep. Okay.
Right.
I'm ready. Let's find that giant keratoisis.
Tomorrow, when we get deeper,
Yeah, maybe.
we'll get the keratoisis.
Maybe not until we get further south. The way things are shaping up.
No, I think we just need to be deeper. I wonder if anybody's got the I Am a Rock song on there. You should be playing that. Well, we climbed this little knoll and look what we have. Look what we found.
There's a large tubular sponge over on the right.
Oh, wow. Ooh, that's a big one.
Yep.
Oh, wow.
Have a closer look at it.
I think this is the one we've seen a few times now today. Would you agree, Sarah?
Mm-hmm.
No,
Oh.
it looks different.
That's why I asked. That's good. Oh, this is the one with the feet.
Which is
Closer.
Thanks.
That's different.
Can you turn down the brightness a little? Yeah.
Oh, this feathery hydroid fuzz.
It
Interesting.
looks like it. It's difficult to say because roselids are generally soft, and
Yeah.
it would be hard for it to stand up straight like that.
I see.
Yeah, I don't know. Chris is watching though, so he'll be happy to see this in 30
Alright.
seconds.
I know that attachment type is helpful, I think.
Thank you, I think that's good.
All right. Is that sea star in the upper right different?
That looks like the one that was underneath the.
Oh, under the Paragorgia. Yeah.
It almost looks like a hysteria.
Yep.
This is a small one.
I don't think we need to go closer. Just right now, I'm doing the sea star.
You're doing the sea star?
Yeah. Thanks. Very spiny.
Well, Chris thinks it's a roselid.
Oh,
Okay.
that's an interesting one.
That could be like the Bachydoryx or something.
Okay. Let's move on up this ridge, see what we see.
So
Chris
what was
Kelly,
it
if Chris is listening, I want some credit for pushing us up the ridge.
What was the decision on this sponge?
Russelidae.
What, Rossellid?
Russellid,
Another
yep.
Rossellid? Okay.
Is this another tumbled over Paragorgia or the one we were looking at before? This is a different one, isn't it? Huh. Looks like it doesn't have quite as many urchins, but it does have urchins on it.
But it's been stripped bare, though. You can see the white
Yep.
skeleton.
Yep.
Yeah. Huh. Wonder what causes them to tip over. Well, I know that sometimes they get too big for the rock that they're on,
Sure.
and
The
that
whole rock tips.
them over. Yeah.
Alright, ROV.
But I also wonder if sometimes the fish knock into them, knock them over. But yeah, that's getting urchined.
Oh, there's a sea star.
But see, the rest of
and
the
they have a
urchins
steer
haven't
it
come
is-
charging down there to get
Yeah,
on
not
that.
yet. Yep.
So, oh, we should look at that star that's
Yep.
on there too.
Two of them.
2 stars.
So I think this is what we were just imaging, Sarah. I think. Yeah.
Those look like more like the Eva blossomates.
That's typical of the hysteria.
There's 3 of them, 4 of them.
Snacking on corals.
Oh.
Yep.
There's more. Yeah. Get that one that's on the bear stuff because it's got a nice angle too. Oh yeah. What's that,
Large
shrimp?
spines are usually indicative of hysteria of the scapula,
Huh.
but I don't know how far they range.
That's okay.
There's another one of those asteroides off to the right.
To the right.
Yeah, if you zoom out a little, you'll see it. A little more. There.
Oh yeah, tucked down and yep, I see him.
One thing that looks most like to me is True Master, but again, the depth is pretty deep for asteroids.
Yeah. Oh,
See, these corals have a love-hate relationship with the echinoderms. They love the brittle stars, not so fond maybe of the urchins and the asteroids. Okay, I think we're good to move on. This is interesting, just a note, the sponge on the lower right, you can see a lot of brown and a little bit of white. So the brown is the dead part of the sponge and the white is the living part of the sponge.
Nearly all dead.
Chris
Yep.
says he's glad you're cracking the whip.
All right, onwards and upwards. What's going on? Is this a sponge growing around the base of a bubblegum coral?
Mm-hmm.
Lost Hero says it's like a slow motion horror movie. That's all right. All the other corals are cringing. Oh, there's another one of these fish that's in a better position.
Oh yeah, get a better view of it.
Yeah.
Yeah. Rockfish.
I seem to. Oh, there we go. Ah, Scott. How many do you think I'd know how to control this thing by now?
Oh yeah, good.
I'm going to take a quick zoom
Good.
now.
Yeah.
Run.
Oh, nice shot. That's good. Oh, look at the fin rays from the pectoral fins
yeah, look at
spread
that.
out on the rock like
Yeah.
that.
Fingers, huh?
Huh.
That's nice. There we go.
Oh,
Get
wow.
a better view. Thank you.
Yeah.
It's got some interesting stuff going on top of its head there.
Taxonomy of this group is pretty well worked out, so
Yeah.
we should be able to figure out who this one is.
All right, let's go back to that Paragorgia. I think it was Paragorgia with a, yeah. See the sponge growing all around it, so it's like the somebody settled on somebody else.
Must have been a really good spot.
And you may have overridden my iris. Oh, there, I got my phone.
Oh yeah.
Everybody likes this rock. I suppose it's possible that the sponge was there and it protected the larval recruit of the coral, and it could grow up in the under the sponge. All right. Well, I'll recap here. So almost 6 hours ago, we landed on the bottom here of Yomie Seamount. On the north end of Yomie Seamount, we landed at 1,520 meters. You're looking at video from the ROV SuBastian, which is being deployed from the RV Falkor, or the Schmidt Ocean Institute. And we are studying the deep sea corals and sponges in this community. And we are currently at 1,327 meters, so we have come up a slope onto this little ridge, got onto the ridge crest, and now we're sort of moving along the ridge crest, and you can see these magnificently large bubblegum corals taking advantage of the current moving across it. A number of these urchins, which we now see are feeding on some of the bamboo coral, any fallen coral. I'm going to stop trying to name things.
Yeah, another rockfish.
A lot of sponges, another rockfish.
Right down in that crevice.
Yep, I see it right there.
Right there, yeah.
Yep.
Yeah, so there are more Paragorgia on this the right hand side, I think. So that would be right. That would be right with the current coming up and over it like that. Yeah.
North face. I think there's maybe another Swiftia right there.
Yeah, and there's another dead Paragorgia skeleton. Yeah.
I think we should take another eDNA sample.
Two of them, actually, probably.
Okay.
This seems to be a pretty dense area.
See now, she wants to take another eDNA. No, on the bottom.
Right, because we were going to do 2 sites on the bottom.
Yep. Yeah, I was just going to say that if I were doing it, I would all want to do it also in places where there weren't such obvious big corals. I want to know if I could also get the small corals. Yeah. But I have no problem taking another sample. So
yeah, this would be a good duplicate.
So on the bottom?
On the bottom, correct. Yeah. Yeah. Meaning, I would want to know, could you get the nodes? Could you get the black corals? Because I wouldn't be doing it just for something.
Right, we discussed that, but we didn't know if they, let's didn't think they were dense enough to worth trying.
Just one left, right?
Yep. Number 4. They're interesting. They're kind of crumbly, so it's hard. But you could take your thumb and move it across, and it would be like sand coming off. But it would erode it yourself. That's what I call it. Kind of like consolidated, like concrete in that sense. It's all very glued together.
We didn't see any white anemones today.
Nope.
No. Okay.
Fired. All right, Niskin 4, YOM 1121. Yep. Got just a few minutes left.
Such a garden. Such a garden. Whew. Yep. Enjoy the view.
Yep.
There are a lot. Get on the ridge, there are a lot.
Yeah. It's impressive.
Classic.
So why are there not more Swiftia up here?
Yeah.
I saw one.
Well, the thing is that, you know, we're at the, I would say we're at the far western edge of the range of Swiftia Pacifica.
There's Boulder here.
So, yeah.
Are you dropping over the side here, J-Rod?
Kind of right now. I'm just trying to get a good setup for the dozen.
I see.
And also flying.
Yeah. It's really broken up into chunks, the bottom here.
You should have come up this side.
Shoulda, woulda, coulda. Now we know. So now we have a greater appreciation for this side, having
That's
come
right.
up the other side.
Hey.
There's all the urchins. We were wondering where they were before.
Yeah, they're all on the up current side, aren't they?
Yeah.
Yeah.
There's one of those large anemones. Can we just have a quick look at that?
Yeah. But see, that Paragorgia is growing against the ground and nobody's on it. Well, that's not true.
Yeah.
It fell over. It's an old one.
Yeah. This is, I think, this
Yeah.
Isoactineris less.
Yeah, as far as we know, yeah, we're going to be diving tomorrow, right? We're going
Yes,
to dive
that
probably about the same time and starting at about 2,200 meters. Somewhere on this seamount, we haven't picked the exact site yet.
Understand.
This is it. Yeah, it's very steep on this side, and there's a lot of Paragorgia and a lot of urchins. Yeah, this is what we saw yesterday.
Yeah, I gotta say, I think that's the Isoactineris.
And they're hard to focus on.
Yeah, I got the focus. I can't get the light though. Maybe because it's so far up there.
Yeah.
Okay. I'm gonna hit the button again, and now I'm gonna try.
Hit it one more time. There you go. Now you're in main.
I think it's lightning. It's way out there. Anyway, that's good. We know what that is. So before you go, though,
Yeah, that's
have
true.
a look at this. Yeah, can you? I hate to ask, JR, can you back up just a little bit? Okay, let's back up and look to the left. There was another downed Paragorgia, but there's still part of the stalk that was stuck to the ground, and there were a whole bunch of urchins climbing it. There it is. You're just getting it into view now in the center.
Yeah, that's there was a. There you go. Yeah. That's look at how big that base was. That was a giant column. I think that's
Yeah, so
like
it's like
the,
broken in two, maybe.
That's kind of like the saguaro cactus. You know, they just get big and big and big, and finally they just get old.
Yep.
And fall over.
Lumberjack urchins.
Yeah.
Yeah, exactly.
Look at that.
Look, they're all over the one down further.
Yeah. It fell in several
Yep.
different directions. Oh, it must have been huge.
Yep.
Look at the size of the base is gigantic. Wow.
There's another one being. All these branches are being attacked by the
Yeah.
urchin. There's one under that pile.
Yeah.
That's really interesting.
But if there's a rampaging storm of urchins going toward this thing, they're moving at a fairly slow rate.
Yes.
Zombie urchins.
A few millimeters a year.
Fascinating. All right, that's good.
Yeah. That's fantastic.
Would you mind imaging the hipposteria behind it? It might be
You don't want to go down that road,
imperialis.
right?
No, we don't want to go down there.
If you, I'm sorry, if you
That
could-
thing?
No, it was
Oh.
on the
Now
coral.
what?
Oh, I thought I did. I thought I did image it.
Oh,
No, I didn't. I thought I zoomed a little bit.
It-
When we first were coming back.
What are we doing?
never mind. It might
got
have been a
5
different
minutes.
hipposteria.
I just have such a hard time watching them feed that I don't like to zoom in on them.
It's tragic.
Yep. There's another one of the fish, not the scorpion fish, but the rockfish.
There's more big bases. Wow. We finally have found the land of the giants on this knoll.
That's for sure.
Oh, there's a sunstar on one.
Oh yeah, let's get. Can we get over there? You think? It's a treacherous place to get through this forest.
There's one on the bird.
Yeah, look at the way that Paragorgia is growing. The rock tipped over,
Yeah, the
and then
rock
it continued to
tipped
go.
over
It
and
looks
it's
like
still
it's
going.
like got a second holdfast, maybe. Look at that. That
And
might
it's down,
be.
and
That might
it's
be worth looking
downwind
at.
of the urchins, so they don't know.
It's definitely got a second holdfast.
Yeah.
Yeah, absolutely. It's smart. Smart coral.
Yep. I think before I said that these don't feed on coral. Yeah, we should
How
have.
did that get up there?
We should have left the bottom earlier.
That's totally bizarre. Is it actually feeding on it, or
Well,
is it
yeah, I don't know.
slurping mucus or something?
Oh, there's nothing's damaged.
Nothing's damaged.
See,
Yep.
But the coral's obviously not upset because the polyps are all out.
Yep.
Just join the breeze.
Maybe.
That one to the right is peaceful.
But he's got a bit of a bulge going, so.
It was a good place to get up and release your gametes.
Yeah. Interesting.
Okay, and then just let's have a turn and look at this coral on the left.
On the left?
Yep, there's a, see that one right down here?
Yeah.
If we could look at it that direction.
I will try.
Thank you. So what's happened is this coral grew for a while on this rock, and it must have got to a size where the rock isn't stable enough and it tipped over. But the coral remained alive, started to grow at 90 degrees upright, but also then laid down a second holdfast to it, more securely fasten it to itself to the rock. That's, I think, what I saw. You could see how bringing a fishing trawl through here would be a bit of a problem.
What's that?
You could see how dragging a fishing trawl through here would be a bit of a problem.
Yeah, exactly. It would be a definite problem here. Yeah, look at that. That's pretty amazing.
It's just like a branch that fell over and started.
Well, I
growing
think
again.
the rock tipped over,
Oh yeah. Okay.
and so then it started to
Yeah.
grow upright, and it's put down a new
Yeah.
holdfast to hold into place.
Crazy.
So that's
I've
some
never seen that
adaptability
before.
right there. I haven't seen that before. I've seen places in canyons where the holdfast spreads out over
Sure.
a lot of the canyon, I think, to consolidate it.
Yeah.
And then new stems can come up, but
Yeah.
not like this.
But nothing like that. This is the whole.
Yeah, go ahead, Bridge.
Should I carry on with my lateral movement?
Like
Yeah, that's fine.
Alright, I'll keep on going.
the holdfast is generated out of the axial tissue along the side of the axis. It's crazy.
Interesting.
Yeah, it's a nice shot there.
Yeah. That's encouraging. It's interesting, like you said, you know, it apparently did it quickly enough that the urchins didn't take advantage of it first.
I got a hunch that the coral basically has to land on the urchin for the urchin to get the urge to start feeding on it. Otherwise,
Well, I
they
think-
just keep grazing away on the rock surface.
Yeah, J-Rod, you asked before, the urchins can't climb up the coral, so here's a perfect opportunity where it could, and apparently that's
But
not
it
the
didn't.
case. Yeah.
Yeah.
So I wonder what the conditions are.
I don't think it even smelled it.
Well, I guess we're pretty much at the end. We do have to stow that fiber before we go, so throw that into your time.
You want to go ahead and do that now?
Sure.
Well done.
Stow the fiber, yeah.
Or pow, as they say in Hawaii.
What did they say?
Pow.
Pow?
P-A-U. Pow. Or pow. Everything's done. Finished.
Oh, I thought you said well done. We're done. Yeah.
We're done. We're pow.
You want to keep your stuff, use your stuff. You don't have to use
And we have a lot to do after dinner. Just in case you wanted to know. Well, I think we know what the shallow stuff looks like now. 2 seamounts in a row, basically the same stuff. Just that we didn't get the concentrations the way we did until we got to the right side of the.
Yeah, but on the
feature.
way, the Swifty were different, the black corals were different.
Yeah.
What else were we
I
seeing?
mean, a
There's
lot of the echinoderms are different.
and some of the sponges, we have different sponges
We
today.
got some sponges we didn't have yesterday.
Yeah.
Yeah. But I think that this comes back to the Apache story. The Apache-ness business, basically.
ROV Bridge.
Yeah, go ahead, Eric.
Hey, I'm just handing over to Jason.
Our last activity here on the bottom is
Go ahead and,
we have a fiber that's been coated in manganese, and it's basically being exposed to the bottom water for the entirety of our dive. When we first arrived on the bottom, we pulled it out of its quiver, and now it's going to be put back in its quiver so it's not exposed to water on the way to the surface. And in this way, we'll be able to learn about the nutrient concentration of the water down here by the corals. But then we're going to get ready to recover the vehicle, bring it back up to the surface, so that'll be the end of the video that we've been showing you, and we really appreciate you tuning in. Glad you're enjoying it. Thanks for your questions and observations. We're happy to try to answer as much as we can. And if the weather cooperates and all the technical conditions cooperate, we'll be doing this again tomorrow. A little bit south of here, still on Yomie Seamount, but we'll be even deeper, starting at about 2,200 meters.
Steal all your stuff because you're falling asleep at 3 o'clock.
So you can check us out on the YouTube feed or on Facebook, or you can go to schmidtocean.org. Where there'll be links to the live video, and there'll be all kinds of blogs and video updates and some information on the mission overall and about the scientists that are out here, the engineers and the crew and so on. So check it out. What time do we dive tomorrow? probably something like, let's see, 2,200 UTCs, so you can figure that out wherever you're living.
I'll take this back upstairs. I went to Amsterdam for a meeting, so I got a pound.
Oh.
So I put a net in, lives in Amsterdam, wanted to get all over the bloody city until we finally found a store that sold them. So now I have one with the European.
So he wants to stay like right here on this position.
Pretty good deal.
Oh, okay.
All right.
We're gonna figure it out. Just tell me.
We need to have a quick meeting.
Meeting about where we're going to go tomorrow.
Yeah.